स्वाभिमान हरी भरी हरियाली खिले सभी साथ में बोल रहे हैं सभी साथ में बोल रहे हैं പാടും കിളികളിൻ നിറവാർന്ന ചില്ലയിൽ താളത്തിലാടി പണ്ടൊരു നാൾ വാസന്ത സൗഗന്ധം അലതല്ലും ചിത്തത്തിൽ മഴമേഘങ്ങൾ മുത്തമിട്ടു ഓർവരമീ മണ്ണു തരളിതയായി ആഗതമേകുന്നു നിറചിരിയായി
Thank you, dear students, for the melodious welcome. Most Honorable Vice Chancellor, University of Calicut, Professor P.P. Ravindran, the Chief Guest of the day and the President of the function, Professor Barbara Zeeb, Dr. Liz Rylett, Executive Vice President, IPS, Professor Om Prakash Dhankar, Vice President of IPS, Professor P.P. Pradyumnan, Senior Professor, Department of Physics, Syndicate Member, University of Calicut, Respected Professor C.C. Harlal, Head Department of Botany, Dean, Faculty of Science, University of Calicut, Respected Organizing Secretary, Professor Josti Puttur, IQAC Director, Department of Botany, University of Calicut, dignitaries on and off the dais, eminent scientists, research scholars and students, a very warm welcome to the inaugural ceremony of 18th International Phytotechnology Conference, jointly organized by International Phytotechnology Society and Department of Botany, University of Calicut. I welcome you all again. Now, moving on to the inaugural ceremony, I hereby invite Professor C.C. Harlal, Head Department of Botany, Faculty of Science, to welcome the gathering. Sir, please.
Good morning, everyone, and welcome to the International Phytotechnology Conference, which will be held in the Calicut University campus from 22nd to 24th of this month. In fact, it is a happy occasion for all of us, as the conference which will be inaugurated today is of a serious one. All of us know that phytotechnology, it is a proper amalgamation of the basic science with the latest technological interventions. It has gained so much of a significance in recent times due to ever-increasing issues in the area of environmental system science. As head of the department, I am here to welcome the dignitaries who are here for the inaugural session. We have with us today Professor Barbara Zeeb, who is the President of the International Phytotechnology Society and a faculty with the Royal Military College of Canada. She had technical difficulties in coming over here in person. And on behalf of the University of Calicut and on behalf of the Department of Botany, I welcome Madam to this function. Today, this conference will be inaugurated by our Honorable Vice Chancellor, Ravindran Sir, a senior faculty, a senior professor from the Department of Chemistry. Ever since he assumed charge of the Vice Chancellor of this university, he has been helping us in abstracts concerning this program. And on behalf of the International Phytotechnology Society and on behalf of the Department of Botany, I welcome you, sir, to this function. And the Department of Botany in particular, I welcome you to this function. Professor Om Prakash Dhankar, another executive committee member of the International Phytotechnology Society. He is not new to the Department of Botany and to the University of Calicut as a whole. He had been with us on several occasions, and he is representing the Phytotechnology conference today. And on behalf of the entire Botany fraternity, entire university, I welcome you, sir, to this function. We have another veteran professor, Professor P.P. Pratyumnan, a senior professor from the Department of Physics, who is representing the syndicate of this university. He has been associating with all activities of the Botany department, and his services to the department is commendable. At this outset, sir, I welcome you to this august occasion. Welcome you, sir. I would like to, rather than welcoming, I would like to congratulate and thank my own colleague from the Department of Botany, who has taken so much of an initiative in making this event materialized in the University of Calicut. I know that for the past 10 months he has been with this event, and finally it has come to a reality. Rather than welcoming you, sir, we from the entire Botany fraternity and the campus community, we congratulate you, we thank you, and as a matter of formality, I welcome you to this function. This conference is being attended by good many researchers and faculty members from outside India and also from various states of India. I don't want to name them one by one because they will be having their own sessions. That time they will be having detailed introductions. So at this point of time, I will welcome all of them to this function. Also this program, this inaugural session is being attended by statutory officers of this university, heads of various departments, deans of various faculties. And with one word, I welcome all of them, along with the researchers and students from various departments. I wish all the best to this conference, and with a final word of thanks and appreciation to my colleague, Dr. Josti Putur, I conclude. Thank you.
Thank you, sir, for the warm welcome. Now, moving on to the next session. As Professor Barbara Zeeb is not able to join us in person, but she has sent us the presidential address with love. So let us hear her thoughtful words.
Good morning, everyone, and welcome to the 18th International Phytotechnology Conference in beautiful Kerala, India. My name is Barb Zeeb, and I'm very sorry to not be able to attend in person and meet with you all. I work at the Royal Military College of Canada, where I'm a civilian. Good morning, everyone, and welcome to the 18th Good morning, everyone, and welcome to the 18th International Phytotechnology Conference in beautiful Kerala, India. My name is Barbsy, and I'm very sorry to not be able to attend in person and meet with you all. I work at the Royal Military College of Canada, where I'm a civilian professor and have been running a phytotechnology research laboratory for over 2 decades. The Royal Military College of Canada is a small university where Canadian officer and naval cadets attend to receive their bachelor's education. We also have a large graduate school with mostly civilian graduate students. Unfortunately, due to current diplomatic difficulties between India and Canada, I was unable to secure a visa, and hence neither I nor my students can attend this year. This year's conference theme is Phytotechnologies for Sustainable Environment and Food Safety. I do think it is very fitting that the conference take place in India this year, as there is so much excellent phytoremediation research being conducted in India right now. We see evidence of this in the many published research papers in high-level scientific journals. The IPS was established back in 2006. Our mission is to promote research, education, training, and the application of all technologies that use plants to deal with problems of environmental contamination, carbon sequestration, etc. We are a non-profit, worldwide professional society open to all researchers, but also practitioners, regulators, and site owners. Essentially, anyone who wishes to promote a natural way to deal with environmental problems is welcome. We have an executive board and hold elections every 3 years. In the spring of 2023, I had the great honor of being voted in as president of the International Phytotechnology Society, taking over from Dr. David Sau, who contributed so much to the IPS and held this position through the very difficult COVID years. Our founding president is Dr. Lee Newman. She was a huge force behind the formation of IPS, and you will hear from her shortly. We currently have a wonderful group of officers from the United Kingdom, Italy, the US, and China. Fortunately, some of these individuals are in attendance at the conference this year. I have marked those lucky individuals with a happy face. Please be sure to introduce yourselves to them. My Executive Vice President, Dr. Liz Rylett, will be taking over many of my duties. You will also see a lot of Drs. Marta Marmaroli, Azam Nouri, and Elena Maestri at this conference. In addition to our executive board, we have a board of directors. Again, I have marked those who are able to be present in Kerala with a happy face. You will see Drs. Jason White and of course Dr. Om Prakash Dankar from the US, without whom this conference would not have been possible. The rest of the gang would very much have liked to attend and will hopefully be at subsequent conferences. Several of them will be presenting online. This is our 18th international conference. The first 3 conferences were in the US, and after this, the Society was established in 2006. We then strove to alternate our conference venues between North America and Europe, or Asia, to allow for more international participation. To date, we have had conferences in all of the locations indicated here. So many in the US, 1 in Canada, several in Europe, and also 3 very successful conferences in China. This is our first conference in India, and again, I'm very disappointed to miss it. I believe there is a lot of excellent research being conducted in India right now, and this is definitely a fitting place for the conference to occur this year. I attempted to put together this map of international representation. However, I may have missed someone, for which I apologize, as the participants have been changing even over the past several days. One thing I do want to point out is that we have excellent student participation this year. This is really important, as it's the students who actually do a lot of the real research, while the professors spend more time seeking funding. The on-the-ground organizers, led by Dr. Joseph Pathur and his team, have put together a wonderful program for you. You will shortly hear about and from the Milton Gordon Award recipient, followed by a keynote presentation, and then these various sessions spread between 2 different halls. Don't forget, we also have a poster session, a closing keynote, a special career session for students and phyto scholars, and of course an awards ceremony. Again, I apologize for not being able to join you in person, and I wish you all a very successful and very stimulating conference. Thank you.
Thank you, Professor Barbarazi. Now, may I request Dr. Liz Rylett, Senior Lecturer, IPC Executive Vice President, for the remarks. Ma'am, please.
Hello, everybody. Thank you so much for coming. It's wonderful to see so many people here. I'm scanning the room. And how delighted I am to be here in India. As Barbara mentioned earlier, there is some fantastic phytoremediation research coming out from this country, and it's really, really a wonderful chance for me to be here and to meet some of you in person. Please introduce yourself to me if you get a chance. I'd like to meet everybody here, if possible. It's also an absolute delight to see so many women here, because these men, frankly, they need our help cleaning up the pollution. So welcome, especially to the women here. Thank you very much. I look forward to an excellent conference with some exquisite science. Thank you.
Thank you, Professor. Now, I wholeheartedly invite the distinguished co-convener of the program and Vice President of IPS, Professor Om Prakash Dhankar, Stockbridge School of Agriculture, University of Massachusetts, USA, for stating introductory remarks. Sir, please.
Thank you, Ria. Respected Vice Chancellor, Professor Ravindran, Professor Pardyuman, as syndicate member, and also the Dean of Sciences and Head of Department of Botany, Professor Harilal, and of course, a conference co-convener, Dr. Jose Puthur, and distinguished scientists, dear participants, a very good morning to you all. Namaskaram, Vanakkam. I learned this word in the last 4 years, only one Malayali word I learned that. Thank you. So it is my absolute honor and privilege to welcome you all on behalf of the IPS and also to the 18th International Phytotechnology Conference here at this very beautiful and lush green campus. It's really, really beautiful campus that I've been enjoying, the botanical garden and all these areas here. And thanks to the botany department maintaining one of the best botanical garden in India. The international, as Professor Barbara Jeeves, she mentioned that International Phytotechnology Society organized this conference annually, and every alternate year we try to rotate off from the North America to other countries. So it has been to several other countries like Italy, Serbia, China, and Greece, and many others. And we've been trying for many, many years, maybe 10 years, I think, when Jason was the president of the society, to bring this phytotechnology conference in India. Finally, we succeed. We found the right host at the University of Calicut to host it. And we are thrilled to have it here now. And also, so this is the first time it's happening in India. And we are delighted to and appreciate your enthusiastic participation. And also, I'm thrilled to report that there are more than there are 210 participants, registrants, and also those are from the 19 different countries, and many are students, speaker students, and there will be 77 talks, platform talks, and around 26 or 27 posters, and so each bringing those the wealth of knowledge, experience, and passion for phytotechnologies. Also, this conference is a platform to foster the meaningful conversation, collaborations, and exploring the innovative solutions to some of the world's most pressing environmental challenges. I'll be talking about some of the logistics here. First thing is the session chairs, different sessions, and some of you might not be aware of that or may not have received the email, but please check the program that you are session chairs and mark your calendar for those days. And also we are requesting all participant speakers, please be respectful of the time. It will be strictly enforced because we need to manage the program smoothly. For all the faculty and scientists, the talks are 20 minutes. And for students, there are 15-minute talks. And session chairs, please ensure that all students get the equal amount of presentation time, because there will be a competition for students, best posters and best talks. In the oral presentation, there will be 3 awards, first, second, and third awards, and same thing in the poster category as well. So students, please make your presentation very nice and present in a way, and so that you can be competing for the award. So judges will be going judging those talks and as well as posters. Also, session chairs, please, if there is any conflict that you cannot chair the session, let us know. Me and Josh will try to accommodate other needs, okay? And for the parallel sessions, the one session will be in the Senate Hall. The second parallel session will be in the adjacent building, in the physical education building. You walk through the food court and then go behind that, and that's on the second floor, Senate Hall. That's the second lecture hall will be. Maybe it takes a minute to go there, and also the poster presentations will be there as well. So now for the next 3 days, we will have great science, networking, and enjoy the great hospitality of the beautiful and also the most delightful cuisines of the Kerala here. And once again, I extend you warm welcome to this IPS meeting, and hope you will have a memorable time here and nice science. Thank you very much. Jai Hind.
Thank you, sir. Honorable Vice Chancellor, Professor P. Ravindran, belongs to the Department of Chemistry, University of Calicut, who is earlier entitled the position of Dean, Faculty of Science, and Head, Department of Chemistry, University of Calicut. Now may I request Professor Dr. P. Ravindran to officially inaugurate the program by delivering the inaugural speech. So please.
Professor Harilal, the President of the function, Professor Barbara Z, Professor Elizabeth Lucy Wright, Professor Om Prakash Dhankar, Professor Pradyumnan, Professor Josti Putur, and all the distinguished dignitaries and delegates, my colleagues and researchers from all over the world, students. It's a great pleasure to welcome you all to Calicut University. On behalf of the University of Calicut as Vice Chancellor, let me extend a warm welcome to all of you for these 2 days when you are here. I also take this opportunity to congratulate the Department of Botany in organizing this International Phytotechnology Conference. I am a chemist, so bear with that. And on phytotechnologies for sustainable environment and food safety, and particularly I congratulate Professor Josh Putur for taking a lot of effort in organizing this program. Well, it's a great opportunity to listen to several of the emerging technologies, phytotechnologies, particularly on sustainable environment and food safety, one of the major themes that globally is becoming more and more important. We all know that our environmental issues have been on the rise in the last several years. We all know our environment is an equilibrium system, and we have been pushing it from every side, and there is always this worry that the developmental burden on environment will not be tolerated any further. So it's very important that we should find alternative technologies. We need to develop ways to communicate the problems with the society, find new technologies for the survival of this life system on Earth. So it's a great thing that we are organizing this wonderful seminar here. We have to learn a lot from nature. And I always used to say that there is no technology which matches with the photosynthesis or the oxygen transport systems in biology. My friend who is teaching bio-inorganic chemistry is sitting there. And so it's very important that we should learn from nature. As organic chemists, people used to say that we will never be able to use water as the synthesis solvent for organic chemistry, but we see that nature always uses water as the medium for organic synthesis. So it's great that we have a lot to learn from nature, particularly when we are being threatened with several issues with respect to the environment, particularly the CO2 emissions, the global warming, and all other issues that affects our atmosphere, our lithosphere, or our biosphere, and all other these spheres of life. So it's a great opportunity to all of us to get connected, being here on this campus, talk to each other, and also I'm sure that this will inspire several youngsters to take up a career in this area in phytotechnology. And thank you all for being here once again. And I formally inaugurate this program. Thank you very much. Jai Hind.
Thank you, sir, for the most inspiring words. Now, I would also request our Honorable Vice Chancellor to officially release the book of abstract of IPC 80. Sir, please. Thank you, sir. Moving on to the next session, I humbly invite Professor P.P. Pradyumnan, Senior Professor, Department of Physics, Syndicate Member, University of Calicut, to deliver the felicitations. Sir, please.
Good morning, one and all. Honorable Vice Chancellor, Professor Ravindran, Professor C.C. Harilal, President of this function, Professor Barbara Sieb, Professor Lucy Relot, Professor Om Prakash Tankar, the co-convener of this program, Professor Jose Puttur, Head of the departments, such students and faculties, and dear friends. First of all, I congratulate Department of Botany and particularly Jose Puttur for taking pain for organizing a international conference, Phytotechnology, on phytotechnology. And the main theme of this particular program, I am very happy to hear that phytotechnology is for sustainable environment and food safety. So formerly we know that this food security, now particularly food safety, because we know that now our environment is polluted by us, and the remedy is also coming from nature, that is they are plants, and those technology we can properly utilized, then in one way we can be more safety in nature, in this particular earth. So I am very happy that many people are participating in this particular conference, and also the deliberations may help our new research students and scientists in this particular field to take their career in a higher stage, and also the new technology may be evolved from this particular conference, and that may help this particular field of phytotechnology. And I know that there are several techniques now people utilizing to eradicate and also make our environment very pure. But at the same time, the sludge from out of that is also making our environment more and more dirty. So at this juncture and during this period, definitely we have to go for new technologies, and particularly how nature is taking this particular environment is more clean and pure. So I am sure that this conference will definitely enhance the knowledge in this particular field. Wish all success for this 3 days conference. And I also thank the organizers for inviting me for this conference. Thank you all.
Thank you, sir, for your motivating words. Now I would like to call upon Professor Jose T. Puttur, IQAC Director, Professor, Department of Botany, University of Calicut, to propose vote of thanks. Sir, please.
Very respected Vice Chancellor of University of Calicut, Professor Ravindran, other distinguished dignitaries on the dais, very senior professors, scientists, junior scientists, participants of this conference, head of the departments of various departments, and other guests. A warm good morning to all. As organizing secretary, right now I am very happy because we are going as per schedule now. 10:45 exactly we will be winding up the first session. So that is one of the concern of the organizing secretary. So in another 5 minutes we will be concluding the first session. Now today we have the most awaited International Phytotechnology Conference being held in our university for the first time in India. And our university has the privilege to hold it in our university. That is the most happiest part of which I feel. And the planning of the same has been started right from 22 when we were organizing the South Zone ISPP meeting here. Om was here, and that was the first time he told that we will organize, if we are willing, we can bring IPC over here. And in fact, he had sacrificed his own home state. He was from Haryana. He was planning to hold it in his university where he did his master's, I think, Rohtak. Then he changed his mind. So that much he has fell in love with our university. So now, also, as already Om has told, this is 18th IPSC. We had all other in different parts of the world, and now we are holding the 18th one. And when we thought of bringing this conference over here, the greatest happiness I felt was that we get all stalwarts here, scientists, very senior and junior scientists here. And there was only one intention before me as well as the department, that our students, our university botany department as well as the allied science students will get opportunity to interact with these stalwarts. That was my greatest, or our department's greatest wish, and that has been realized, and I'm very happy about that. Now, we have released the abstract book. This was done formally. Don't expect that you all will get an abstract book. Everything is online. We want to go green. Just scan your QR code here. You will get your abstract book over there. And Wi-Fi is provided over here, so you can easily make available the abstract book on your phone or lab, whatever it is. Now, another wish I have is that we should have, and I am sure that we'll have good deliberations over here today, because as I had already told, many people who are working in this phytotechnology line are here. In fact, when I was discussing about this conference to my colleagues, they were telling me or asking me, what is the technology in plants? Because they all expected only engineering and technology. But now we are making the people aware that there is a lot of technology to be done with plants, and that may be the more sustainable. And tomorrow you have to depend on plant for a phytotechnology in order to save our Mother Earth. And just I want to caution you that, as I already told in the beginning, we need to keep up time. As already Om told, that is the importance of our schedule. We will have a bell after the sessions when you are for the tea or the lunch. We will just have a school bell, just like a school bell. It will be rung, and that will just to remind you to get back to our sessions. Now I come to my duty is to express a vote of thanks. Now let me come to first our Vice Chancellor. He was so supportive in organizing this conference. First of all, when I discussed with him whether we can hold this conference in Senate Hall, normally this hall is not given for these conferences. But he said, yeah, this is an international one. We will organize it in the Senate Hall because this is only for Senate meetings, academic council meetings generally. But he readily agreed to that. And moreover, he gave generously funds to make sort of a lot of repairs, and the walls you see all are very clean, and everywhere it is clean over here. And that is the generous funds what he has provided. So, sir, thank you for that. And thank you for gracing this occasion, inaugurating this conference. In the absence of Barbara, the president of the society, let me thank her also. She was very desperate to get to this place. Unfortunately, due to the present relations of good relations of India and Canada, she could not reach to this place. You all know what is happening now. But I think these things should not be taken to science. Personally, I feel. But anyway, it has happened, and her student, and in fact, her husband also was wanted to accompany her. All three got stuck up in Canada. And because of the same good relations, another 5 got stuck up in China. So that is anyway bad luck. So in her absence also, I am proposing vote of thanks to Barbara for her deliverance of the lecture, what she has already done here. And also I thank Elizabeth, for her taking the shoes of Barbara and compensating her place on the dais. Thank you, Liz. Now let me thank our captain, Professor Harilal. He is our captain of our department and also the Dean of Science. And he is the convener of this program also. And he has been very cooperative, supportive to this program, and always promoting me. Yeah, you go ahead. I am here. We will see whatever happens. It will be done. So thank you, Dr. Harilal, Professor Harilal, for your support. And coming to our very beloved professor from the Governing Council. Maybe the syndicate, some of you may not be knowing, especially those who are from abroad. Here the syndicate means it is the Governing Council. So they have a lot of power, in fact. That is the thing. So we have Professor Pradyumnan, and he is as he is from science, he is again very supportive to us. And thank you, sir, for your graciously coming over here. Now, at this occasion, I also should thank especially Engineering and Physical Education. Director of Physical Education is here. Sakir is here. And in fact, they have generously donated all their place. You will be enjoying the beauty of that big stadium from their building. And for 3 days, they have said, whatever way you can use the building. So I thank you, Dr. Sakir, for giving us space and all that things. And engineering has supported in doing all this work. And now I should thank our different various sections. I am not naming because of the shortage of time, almost 10:45 now. So all sections has done a good support to our conference. Thank you all. And now I come to the biggest task of you participants. You have made this conference success. You people have traveled from so far wide. US, I think 19 countries home, 19 countries. And taken all the trouble. And Mexican is here. He was telling me that it is almost 2 days journey because their time is reeling back. So even taking all that pain, he is here. So thank you participants for taking an active participation. Thank you all. And if I missed, I'm sorry. If I missed anyone, my faculty members, my colleagues are there. They are all supportive. My students are there from the department. They are very supportive. Today you will see their fantastic performance on the stage during the cultural event. And I thank you all for your support given for this conference, along with my whole faculty members. So thank you all once again. I sharply stop at 10:46. Thank you.
Thank you, sir. Now let us break for the tea. The tea has been served outside the Senate Hall. All are requested to be back by sharp 11:10 a.m. Thank you.
We have to manage, sir. Otherwise, the
One thing I found is you have sacrificed
Those looks good on the plants, right?
You should have some beauty for the place also.
Kindly note, there is an instruction. All the participants and the presenters are requested to submit their presentation in the registration desk. All the participants and the presenters are requested to submit their presentation at the registration desk. Safety. Now, this conference is a call to action to reminder that sustainability is not just an aspiration, but it's a responsibility that we all share for the future of our planet. Together, we are here to shape a future where development is harmonious with nature, equitable and sustainable. So let's embark on this transformative journey together. Now, before moving on to the technical session, let me introduce the Milton P. Gordon Award. Now, this is an award which is presented as a token of appreciation and love by the IPS to a leader who have shown excellence in the field of phytoremediation research in honor of Milton P. Gordon, a pioneer in the respective field. Now may I invite Dr. Lee Newman, past President, IPS, to present the Milton P. Gordon Award.
Hello, everyone. Although I'm not able to be attending the conference in person this year, I am still highly honored to have been asked to give this presentation introducing the 2024 Gordon Award winner. However, before we get to the current winner of this prestigious award, I would like to give a little bit of background about Milton Gordon himself. Milton Gordon was a professor at the University of Washington from 1959 until his retirement in 2003. In 1960, he helped found the prestigious journal Biochemistry and was an associate editor for the journal for 31 years. With his colleague Eugene Nestor and others, he was one of the pioneers in developing the use of agrobacterium for plant transformation and making genetically engineered plants. Hello, everyone. Although I'm not able to be attending the conference in person this year, I am still highly honored to have been asked to give this presentation introducing the 2024 Gordon Award winner. However, before we get to the current winner of this prestigious award, I would like to give a little bit of background about Milton Gordon himself. Milton Gordon was a professor at the University of Washington from 1959 until his retirement in 2003. In 1960, he helped found the prestigious journal Biochemistry and was an associate editor for the journal for 31 years. With his colleague Eugene Nestor and others, he was one of the pioneers in developing the use of agrobacterium for plant transformation and making genetically engineered plants. When NASA's space shuttle started doing biological experimentation, he was one of the advisory board members helping guide what research would be conducted on the space shuttle. Milt Gordon served as one of the founding board members of Calgene Incorporated, which was most noted for its production of the genetically engineered Flavor Saver tomato. But it was in 1982 that Milt started his research in phytoremediation, and it is for this that we honor him today. Milt Gordon did work in the laboratory, in the greenhouse, on test sites, and was an active team member when we were actually installing phytoremediation as a cleanup remedy on contaminated sites. While Milt's generosity to the students and postdocs working in his lab was legendary, What wasn't known or well known was the amount of time that he spent giving to other people who were less fortunate than him. What we see here is a picture of him working on a field site in Mexico with Habitat for Humanity, along with the former president, Jimmy Carter. Unfortunately, we lost Milt all too soon when he passed away in 2005. In 2007, those of us who had worked with Milt Gordon, who had known him, respected him, and admired him, came together to create the Gordon Award. Each year, we honor a member of our society, a member of our phytotechnology community, who show the traits that we so admired in Milt: a love of teaching, a love of research, a willingness to reach out and help others scientifically and personally, and a dedication to promoting phytoremediation in all aspects, from laboratory to bench scale to actual application in the field, cleaning up the problems that we face in our environment. Over the years, I have been honored to present this award to some of the giants in the field of phytoremediation: Jerry Schnoor, Alan Baker, Larry Erickson, Rufus Cheney, Jaco van Grunsveld, Nelson Marmarelli, Michel Mensch, Stanislaw Boronsky, Barbara Zeeb, Jean-Louis Morel, Joel Birkin, and last year I was incredibly honored to receive the award myself. This year, it's all about our latest recipient. She got her PhD in plant genetics and biochemistry at the John Innes Center in the UK. She has spent her career studying the mechanisms of plant-induced stress following exposure to xenobiotic compounds, the use of genetic engineering and synthetic biology to develop plants that are better able to degrade organic contaminants, and manipulating metal uptake, transport, and safe storage in plants. This year, I am incredibly honored to present the 2024 Gordon Award recipient, Dr. Liz Rylott.
Okay, you should wave if you can't hear me. Okay, almost telling me to wait.
Thank you. And now the award will be presented by on behalf of Lee and others, and Barbara. It will be presented by Dr. Jason White, the past president of the IPS Society. Thank you. To Liz, please.
Okay, enough chit-chat and officialness. Let's have some science. Oh, I've got a prodder as well. So, as I said, I work at the University of York in the UK. My work covers environmental pollution of organics, but also metals. So I do a lot of work on the metals. I'm talking later in the week on that. Today, I'm going to focus on environmental pollution of organics. And for that, I'm using exemplars, explosives, okay? So explosives might not seem a big common pollutant out there, but it's an exemplar. If we can learn how to clean up this pollution, which we've received some funding over many years, particularly from the US Defense, to look at this problem, then we can roll this out to a number of organic contaminants that I know are big issues globally and here in India. So let's get started. I've got a picture here, which is, on the one hand, is a lovely woman here keeping the bombs nice and polished, because you wouldn't want a dirty bomb to drop on you. You want a nice clean bomb when it blows your house to pieces. And then a picture on your right-hand side there of the Somme in the First World War, and just a graphic reminder of not just the horrific pollution, but the absolutely horrific loss of life. So let's lift the mood a little. How do we move forward? That one? Hey, we're in. Okay, so the two pollutants I'm going to talk to you about today, these are partners in crime. We have something called RDX and TNT, okay? So if you heard of TNT, yeah? Maybe not RDX so much. Some nods in the audience, thank you. So these compounds are not great. They're co-contaminants, so you find them together. They're not found in nature. In the case with many of our environmental pollutants, we've made these all by ourselves. We're really clever, but they can't necessarily be broken down in the environment. So these are hanging around for decades, if not hundreds of years in our environment. So we're going to drill down into the problem with these explosives. So the problem with RDX, this one, it very readily leaches into the groundwater. So it's causing big problems in the US military on their training ranges, because they want to keep that. They want to use live munition, but they want to contain it. This, if it gets into the groundwater, is not good. It's toxic, and it's a significant issue. And so here you can see, did I miss a slide? Okay, thank you. That's great. So the big problem with it leaching into the ground is that you've got your squaddies there on the US site. You move the slide. I'll just wave. You move. Oh, okay. We'll work it out. No, this isn't working anything. Okay, so it rains. It rains, and then the RDX is washed into the soil. Can we have the next slide? Oh, okay. And that washes, in this case, we've got a sole source aquifer in the US where the RDX is accumulating. I can't seem to move any. It's not working with the right flicker. Oh, no. It's a bit slow. Oh, okay. Can you take it back? Okay, that's great. Let's hold it there. And so this sole source aquifer, this is where the RDX accumulates. And it's not good. It's an environmental pollutant. We know that it's a toxin. It's a suspected carcinogen. And in this town, all the people that live there, they take their water from this sole source aquifer. Now, I'm not saying that RDX is necessarily the problem, but they have a higher incidence of breast cancer, for example, the people that are drinking this water, and there are other pollutants in there as well. So environmental pollutants need to be cleaned up. Shall I press? Thank you. The second one, this is a very nasty, toxic, and persistent constituent on our planet. I refer, of course, to TNT that you can see here. It's also a nasty component of rifle ammunition, as demonstrated here by this gentleman. It's carcinogenic, and as you can see from the chemical structure, those nitro groups are pulling the electrons out from the ring, making it very, very difficult to attack that ring with an enzymatic approach. So breaking down RDX enzymatically is a very, very big challenge. It also binds tightly to the humic content. So unlike RDX, which is leaching very quickly through the soil, the TNT is bind tightly. And so we're going to use this as an exemplar compound for phytoremediation of organics. If we can break this down, then we can deal with DDT, dealdrin, other components that we can look at and see if we can find ways that we can break those down in the environment as well. Okay, do you want the next one? Thank you. So what's the extent of the pollution of explosives? Perhaps until today you'd never even heard of explosives as an environmental pollutant, but they are. We often don't have records of them, though. It's not something that countries have wanted other countries to necessarily know about. But in the US alone, where they do keep very good records on environmental pollution, there's over 10 million hectares of contaminated land on their active training ranges, billions of litres of groundwater, and the costs are astronomical. But the costs, should we do the next slide? Thank you. The costs are not really the problem. The problem is we don't really know what to do with it. So where has this pollution come from? In the case of explosives, a lot of it is manufacture. So manufacture during world wars, but still there's a lot of active production of TNT and other explosive compounds, and this leads to environmental pollution. It's unavoidable. Next slide. So after manufacture, can we have next slide? Oops, okay. Back, well, we've got use as well. So bombs, when you use them, they leave remnants of explosives in the ground. And you can see here there's a howitzer shell on your left-hand side there with particles of RDX and TNT that are still live, that haven't been exploded. You can see the pond water, it's called red water. So that is TNT actually in the ground, giving the water that orangey red color. There's so much of it there. So this is a problem when we use it, but finally also when we decommission it. So bombs have got use-by dates. I mean, who knew? Your food in your fridge might have a use-by date, but bombs have a use-by date too. And so after that time, they store them somewhere and then leave them, and they're going to accrue in the environment. How are we going to deal with that? So we've got Obama was going to deal with that. We've got which was Biden, and he was there from the previous time, and I guess maybe I'll keep him there for the next time. So current cleanup strategies, there are a number of different strategies: incineration, landfill, bioaugmentation, composting. These are great and they have their place, very much so. But the scale of this problem is enormous. That's why we want to use plants to try and clean something up on a huge scale. Thank you. So I probably don't need to spend long on this slide. Phytoremediation offers lots of benefits. If we can make this work, we can do a lot of things to really help improve our environment. Okay, thank you. So our mission, ladies and gentlemen, like Tom Cruise, is to remediate and contain, in this case, explosive contamination on military training ranges. That's what we've got to do. And we were tasked with this by funding from the US military, who've been very, very generous with our funding. So the first problem, though, next slide. If we're going to use plants to detoxify these environmental pollutants, back one, we have some problems. So we need to ask these questions and see if we can get around them. First of all, TNT is very toxic to plants, so we can't use plants to remediate it because they're going to die. They need to be toughened up, if you like. And then, if we're going to toughen them up, how are they going to break down TNT? How are they going to have the enzymatic ability to break down those difficult chemical structures in these chemicals? And then finally, it's partner in crime, which is not quite so toxic, but how can that be broken down as well? How can we... The classical genetic approach. This is one of my favorite childhood books, The Enormous Turnip. Do you have an Indian equivalent? what the genes are that are controlling this. We have no idea, okay? So then we have to go through a series of experiments to identify the genes. One of the candidates that came up was something with this really catchy short name. It's called monodehydroascorbate reductase, okay? And it's involved in the schematic of recycling, essentially vitamin C. So you might well be aware that vitamin C is an antioxidant in our own bodies, and plants use vitamin C too. And this enzyme, its job is to make more vitamin C. So why would knocking it out make a plant more resistant to stress? And we were like, we don't understand, we don't quite get this. So we left this one on the shelf for a long time before we came round to look at this gene. Okay, next slide. The first thing we did was find more mutants in this gene. And you can go to existing mutant libraries and find more genes. And we found 2 more alleles in this gene, and we can see that they gave us the same phenotype. So we know that it is this gene that's causing that resistance. So next slide. We also saw this in roots. And then, next slide. Then we ground up the plants and we gave them the substrate to see if there was any change in the activity. And there was. So when we give them that substrate that they're using would normally turn into vitamin C, we can see that they've got less activity. And when we give them TNT, just ground up plant extract, we give them TNT, we can see that there's a decrease there as well. So the plants are doing something to the TNT, but what is it? We still don't know. Okay, this took years. It would take much quicker if we'd known from the start. With the next slide. So we purified the enzyme, and this is a slide really quick to do, but this took us months and months. This was not an easy to produce enzyme. We tried lots and lots of different ways to optimize it. We finally purified it, and there it is, and it has activity to its natural substrate. Okay. So the next step was to give it some TNT. So we gave it some TNT to see what would happen, and it had activity towards TNT, which was quite remarkable. It's got activity. What is it doing to the TNT? So we're measuring this using a substrate cofactor, NADH, which has a change in absorbance at 340 nanometers in a spectrophotometer. So it's easy to measure. And we can see that as the protein is fed that TNT, we're seeing an increase in activity. It's doing something with the TNT. That's really exciting. It's got activity. But then when we measure how much TNT is left over, it's all still there. I was like, well, what's happened? Let's go again. So it's not changed the TNT, but it's used up all the cofactor NADH. We go back in the literature, next slide, and we can find that there's evidence in old literature that MDAR6 and other enzymes, not MDAR6, other enzymes, can catalyze a 1-electron reduction of TNT. Okay, next one. So then we did something called electron parametric resonance analysis to see what was going on, to see if we can find that. So what I'm showing you here is a way that we can see if superoxide is being produced when TNT is catalyzed by our enzyme MDAR6. So at the top here, we have a spin trap chemical, which holds the superoxide for longer so that we can see the signal, if you like. And this is a simulation, what we see here. This is what we would expect to see. So, next slide. When we put the TNT and the enzyme together, do we see something that looks similar? We do. This is really exciting. But then it's very important as a scientist to do your proper controls. So we then take the enzyme and the TNT and we do controls with that. So if you do the next few slides. First of all, we add something called superoxide dismutase, which gets rid of the superoxide. Yeah, next one and the next one and the next one. And we lose the signal. If we take the enzyme but no TNT, we lose the signal. And of course, if we boil the enzyme, we lose the signal. So this is very, very strong evidence that we can see that. And then on the right-hand side, we can see we repeated the simulation and the experiments with a second spin trap chemical. So we're on board. We're trying to work out what's going on with TNT in the plants. So if you do the next step, one final experiment that we did was, is hydrogen peroxide produced in the roots of the plants? So here's some wild-type Arabidopsis roots without any TNT. And the next slide are mutant without any TNT. Oh, okay. Yeah, just click the next two as well. So you can see in the absence of TNT, the roots, we've got a stain here that stains for superoxide. When there's no TNT, the roots don't really stain very brown. But when there is TNT, you can see the wild-type roots stain very brown because there's lots and lots of superoxide being produced by our enzyme. And our mutant, where the enzyme is knocked out, is not producing that, and this is why it's so resistant. So if you go on to the next slide, I can give you a summary of this. So 1 electron reduction of TNT. This is why TNT is toxic to plants. So here you've got MDAR6. And this enzyme doesn't live just anywhere. It lives in the mitochondrion. So the mitochondrion is like, this is a really important part of your cell. This is where all the important biology is going on. This is like Delhi, if you like. Whereas the cytosol is like, no one here from Australia, is there? It's like Australia, if you like. You know, it's the backwater. It's quite diluted. MDAR6 is in a really important hub. Okay, I'm going to get into all sorts of trouble now. Okay, next one. What it does in that mitochondrion is taking TNT. Okay, TNT is coming through into the plant. It's passing up with passive transport straight up into the cytosol and into mitochondria, where it sits. MDAR6 takes it. Yep, keep clicking, go on. And it makes this nitroso radical. This nitroso radical is not happy. It's had an electron stripped off it. It wants that electron back. It gets the electron back. To do that, it uses... So it's taking NADH to do the first step. And the second step, it uses molecular oxygen to scavenge that electron back, and it makes superoxide. Keep going. Keep clicking. Just keep clicking. The next 5. Okay. Then it can do this round in a cycle. It can do this forever. It can keep going round. The TNT is made into nitroso radical that depletes your NADH, which you need. Plants need that. It produces superoxide, which the plants don't want. It causes damage. And this goes round and round and round. And this is why TNT is so phytotoxic. If you keep going, I've got a little bit of kind of cartoon. I think I made it heat up and explode. But that bit, we don't know for sure. But, you know, your mitochondria are dying out. And this enzyme is also targeted to the chloroplasts as well. So you've got a lot of damage. We now know why TNT is toxic to the plants. So keep clicking. Okay, so we published this in Science a while ago. But since then, this research has enabled us to make further gains in understanding how we can develop plants that will be resistant to TNT. So, yep. So there's your wild type. That's what's happening in the mitochondria of the wild types. That's why they look so ill. Whereas on the right-hand side, you can see that enzyme has been knocked out, the gene's knocked out. That's not happening, and the plants are doing really well. Okay, so keep going. So now we know what the toxicity is. How do we work to make plants that are more resistant? How can plants cope? Well, they can detoxify some TNT before it gets to the mitochondrion. They can do an activation step, a conjugation step, compartmentalization, which is a bit like my house cleaning, where you would sweep it under the rug, and they can also transform it. Okay, so let's look at a few of these steps. So here are the roots of TNT, and this is a pretty horrible slide. We've got lots of chemical structures, but don't worry, I'll hold your hand. Next step. Yeah. Okay, so you can see one of the nitro groups here. It becomes a hydroxyl and then amino, hydroxyl amino group and an amino group. This is how plants start to detoxify. They're doing an activation step here. This is putting a tag on this molecule that makes it more water soluble, and the plant can then take it further away and detoxify it. So the enzymes that do this, we have a second step that will go down that way as well. The enzymes that do these are nitroreductases, and in the plant they're called oxophytodianoate reductases, okay, OPRs. So nitroreductase will do this first step. If you can do that first step, you can stop that TNT getting into the mitochondrion and causing all that damage that we talked about. Okay, so can we increase the levels of this? We tried to do this by increasing the levels naturally of their endogenous OPRs, and that's a lot of work. It was a whole PhD, but it didn't make a lot of difference, to be fair. When we bring in a bacterial nitroreductase, which is not under the regulatory control of the plant's own policing system, if you like, the bacterial nitroreductase will do this process much, much more quickly. And you can see these tobacco plants on the right-hand side here. We've engineered them with a nitroreductase from bacteria, and they are significantly more resistant because they can get that first step. They can start to activate the TNT and stop it going into the mitochondrion. Okay. Thanks. The second step is conjugation. It's going to be attached to a sugar molecule. Okay, and that means it can then be taken off and put into the vacuole or sequestered or perhaps stored into the cell wall macromolecular structures, right? So we looked at, and I've got some slides down here which I made really small because I'm really sad because this was at least another PhD and half a postdoc's life gone on this, to characterize, I think we characterized about 8 or 10 different glutathione transferases, glucosyl transferases that will do this step. And there's lots of lovely biology, and it was really, really interesting for other environmental organics, not TNT so much. So essentially, I think I'm saying it didn't really work. It didn't give us that resistance that we were after. Okay. So we put that on the shelf. We thought, well, that's good. We understand more about that. Then we looked at these glutathione transferases. Well, we hadn't really wanted to look at those. We didn't think they would be so important. So the glutathione transferases, yeah, thanks. They do a conjugation step as well. And so what they do is they take TNT and glutathione, stick the two together, and then they can make that molecule more functionalized, and it can be hidden away within the plant. And it makes 3 different conjugates, okay? So it'll do conjugate 1, conjugate 2, and the third one that we're really interested in, conjugate 3. And I guess I can show you here. It's this bit here. We've got a sulfur group instead of a nitro group. So when I spoke earlier, I talked about the 3 nitro groups pulling those electrons out from the ring. We've taken one of those nitro groups off. Well, I haven't. The plant has, all by itself. And this has destabilized the ring. We know that nitrotoluenes, dinitrotoluenes, so not TNT, but DNTs can be degraded. So this is a very exciting step in our odyssey to make plants resistant and degrade TNT. Okay. Sorry, not very clear. So we then looked at the endogenous plants that do this, enzymes, all back at one. So we've got U24 and U25. These were the 2 enzymes that do this. And one of them would make a lot more of that conjugate 3 than the other. So that's what we want more of that conjugate. We expressed this at higher levels in the Arabidopsis plants to see if it made a difference. And it did. They could remove twice as much TNT as the wild type. So this is really promising. Yeah, go again. But we only want conjugate 3. The other 2 conjugates we don't want. We want to take that nitro group and replace it with a sulfur. So we looked at 2 different approaches to do that. And so one way is to look with a site-directed mutagenesis approach. So on the graph at the bottom left there, on the one side we've got U25, an enzyme that makes predominantly the conjugates that we don't want. And on the other side we've got U24 that makes a lot of conjugate 3. That's the blue stripy bar. And what we did was we looked at the active site of the enzyme and we swapped amino acids out 1 or 2 at a time. We were gradually able to show that we could move from one side of the graph to the other. We could recreate that production for conjugate 3. So that was one mechanism, if you like, that would give us more conjugate 3. But a second mechanism was just by coming to a conference like this and meeting someone who was working on Drosophila, who said, whoa, we've got a glutathione transferase I think you should try. And we tried that, and it gave us an absolute bucket full of conjugate 3, exactly what we wanted. So next slide, please. So this work, I'm not going to talk much more about this, but this work then enabled us to use it as a tool to elucidate further the fate of TNT in plants. So I come to conferences and people often talk about, oh, it's been taken up by the plant. Yay, job done. But it's not job done until we know exactly what's happened to that structure, that pollutant that we gave to the plant. Where has it fundamentally ended up? Is it mineralized? Is it tied to macromolecular structures? We don't know. Because this line here, this Drosophila expressing plant line, produces much higher levels of that conjugate 3, we can trace that through the plant, and that's what we've done with these experiments here. So we're looking at how this is being transported and where it's being put, in this case, into the vacuole. And so all of these different structures, we're now in the process of working out how this very complicated jigsaw fits together. So in summary, we know that TNT is toxic, we know why it's toxic, and we know that it can be activated and conjugated to detoxify in the plant. Okay. Yeah. So our next question was about the RDX. This is the co-contaminant that we've got with TNT, the two bad boys, if you like. We think we're We're starting to deal with TNT, but we haven't talked about RDX at all. So how does RDX get broken down? Well, the plant doesn't have any real innate ability to break down RDX itself. It doesn't have any enzymatic activity that really does the job. If it did, then it might start to clear up the mess on its own out there. But we know that military training ranges, which are heavily vegetated, that the TNT and RDX is coming out from the bottom into the streams and rivers. So the best way to find enzymes that are able to break down environmental organic pollutants is to look in a place where we as humans have dumped a hodload of them years ago. So go somewhere polluted and look for bacteria that are breaking down those compounds. RDX is full of nitrogen, so there's a huge evolutionary pressure going on in the soil for the bacteria to evolve the ability. If they can eat the RDX, they've got a little niche, haven't they? They're doing really well. And so that's what we did. We sent the French guy down into the crater there to get some samples. And he found this beautiful bacterium here called Rhodococcus rhodocrus. And that's, you can see it streaked out on the plate. And we then, from that, we isolated two enzymes, XPLA, which is a cytochrome C reductase, a cytochrome C, and a reductase, a reductive partner. And these two work together, and they catalyze the first steps in RDX degradation. We published this, and you can see here essentially what happens is that they do these denitration steps, and then the molecule will start to fall apart. And these downstream compounds, Medina and NDAB that I mentioned, we've done some studies further, and we can show that plants are mineralizing those, although you do see some of these in the environment still, so some bacteria won't necessarily eat them. But we're getting a long way. We're nearly mineralizing RDX. We're getting there, so it's looking really good. So we published that, and we moved on. And then I want to say, here is a summary of explosives detoxification that we put into the International Journal of Phytoremediation that summarizes everything that I've said. So if you want to know more, that's perhaps quite a good place to have a read. And I just want to put my beautiful figure up there because it took me hours to make. Okay, so that's a summary of the explosive detoxification. The next step is, there's no point doing all this academia if we don't work with the industries, the pollutants out there, the other stakeholders, to make sure that our research is applied, that it goes out there and it works, and somehow the pollution is cleaned up from our efforts in part, but other stakeholders, businesses, industries. I can see lots of people nodding. We know how important that is. So can we use these plants? Can we develop plants that will clean up the military training range pollution? So what we've got here is what our wild type plants. We've got a no plant control. That's what MPC is. And we've got plants expressing this cytochrome P450 and its reductive partner. Okay, next slide. Sorry, that's a watering can. That was a bit loud. We watered RDX into the plants because we're trying to mimic a training range and some rain. So it was a very fundamental, simplistic experiment, but it gets the idea. Then we left the plants for 7 days, and then we applied the water again to see if any RDX would flush out as it flushes out on the training ranges. Okay, and you can see the RDX is coming straight out of the pots with no plants in them, and the wild type plants. But our transgenic plants, they've taken this up, they've broken it down. And sometimes you're in the lab and we do physically hug, and we do have champagne, and we do celebrate, and whoop. We have a big stack of tissues as well, because a lot of experiments don't work. But this was a really good experiment. So the next step is to combine these into a soil experiment with our nitroreductase. So I mentioned earlier the bacterial nitroreductase, okay, that will help plants detoxify TNT quickly before it gets into the mitochondria and starts causing all that damage, okay. So we put our nitroreductase into the plants and our XPLAB RDX degrading enzymes, put them all into the plant, okay. And then we did this soil experiment. So first of all, we have unmodified plants, and you can see the devastating toxicity of the TNT and RDX together. Then our next experiment is just the controls, okay? This is just the nitroreductase plants alone, and you can see they're more resistant to the TNT than the first pot, but they're not as good as they could be, all right? And we haven't got rid of the RDX, that's still in the soil. Then the next experiment, is part of the control, it's just the XPLAB that degrade the RDX. They're really good at doing that, but they can't do it in the presence of the TNT because the TNT is killing the plants. But we combine them together, and you can see that we can really, more or less, make these plants almost completely resistant to the toxicity of that TNT and they're breaking down that RDX. So this is really, really good. Okay. This looks like it might work in the field. Okay. But I don't know if anybody knows about Arabidopsis. It's not unlike a lettuce. And if you've seen a tank, it's not really compatible. We can't work in the field with this species. And this is a big bottleneck in phytoremediation at the moment. It's moving from model systems in laboratories to something out there that you could actually grow. They have different genomes, there's complexities, they're big, they grow very fast, but they grow very slow through life cycles. There are lots and lots of problems. So what species did we hit on in the end? We chose switchgrass. Okay? Yeah, that's all right. Okay, so switchgrass is a native plant species. It grows on the training ranges. It provides all year round cover. It's a really, really good system. And when they're out there with their tanks, it can be literally blown off the face of the earth. It'll land back down, splat, and it'll start growing again. This is a really, really tough plant. This is not Arabidopsis, okay? So this is what we're going to use. But we can't transform it. I talked about those bottlenecks. We can't transform it. We don't know how quickly to grow. We don't even know exactly how to grow it. It took us a very long time to work out how to get seeds back from it. So we tested some promoters, and the top image there shows a Gus promoters testing that. Monocot species are very specific and particular about the promoter that drives their gene expression. You can put a 35S promoter in front of a gene in a dicot plant, and they're perfectly happy. You mess with those promoters in a monocot, they're not going to express that gene. So we made a number of different constructs testing different monocot promoters. We made multi-gene constructs, and we had to develop from scratch a transformation methods we were working on wheatgrass and we were not getting anywhere. And then another group published for switchgrass because it's a biomass crop and they were working on a completely different scientific topic, but it shows science coming together and giving each other help. So we were able to transform switchgrass with our genes. Okay. So another thing that is really important to remember about is herbivory and the rest of the food chain. So we've made plants that can take RDX from the leachate. We've got plants that grow much, much bigger than unmodified plants on the TNT. So they look like they can really do, let's roll out these plants really quickly. Now, hang on, let's have a little think. What are we doing here? What about herbivory and the rest of the food chain? We have changed fundamentally some plants here. We're using some GM technology. Are the plants full of TNT and RDX? Is it going to be a problem? Are they going to be toxic? So we did a lot of different toxicity testing, and I won't go into the work that we did here. We did a lot. And the plants contain a lot less RDX than the wild types, and I'll show you some data later. And the feeding preferences, there was no difference. They couldn't taste more or less. I've not eaten TNT or RDX myself. I don't recommend that you try it. But the insects, they couldn't tell. There was no difference, okay? So it's a cursory check, but at this stage, we're just going to do a small-scale field trial under very controlled conditions as well. So we get the go-ahead for the field trials, and this is our design. So we're going to grow the switchgrass. We're going to have a little closed system so we know exactly how much RDX we put in. We're now just working on the RDX. We're not looking at the TNT problem. And we're going to grow the plants there, and we're going to cycle that RDX round with the rainwater, and we're going to see how it goes. So, yeah. So I've just got some holiday snaps next. Let's go. So this is us setting up on the field trials. There's Tony Palazzo, he was there a minute ago, and Tim Carey, who were instrumental in setting this up. And there was me setting some lysimeters up on there as well, and we're getting all of these different plots. And you can see we put a number of plots. This is quite a big site here. It's maybe about a quarter of a mile across in length. And this is in the US, and it was on a military training site, and we had all sorts of issues working with stakeholders again that were not academics. They didn't know what we were doing. We didn't really have a lot of... we weren't very fussed about security per se. I'd say the US military are very fussed about security. But this was on their site, and it was at the end of the drone runway as well. So they were able to take aerial photos of our field site as well, so you can get a feel for it. So setting up something really, really big and doing it from the other side of the pond is also logistically incredibly challenging. We arranged for the RDX to arrive at the military base in the dead of night. They had a helicopter above it, shining a light on it, saying, what on earth is this? We were in trouble several times with the military. The logistics were enormous. We started to plant up the samples, and you can see that's what the plots looked near the start of the experiment. And so on the next slide, you can see the growth of the switchgrass on the plots across the first year. And then 2017, and I'm not sure I'll ever repeat a field trial because you cannot control the conditions like you can in a lab. 2017 was the wettest year on record in that area. And our plots, I think I mentioned, are closed, they're lined. We were growing wheatgrass. We should have been working with rice. You know, we had to really bail this out. At the back of this photograph, perhaps you can see there's the original tank that we had, and then we had to buy a whole load more, much bigger tanks to pump the water off from each plot. And then we had to measure the RDX in each of those tanks, and then we had to chemically destroy the RDX and then rinse those tanks out. And then the tanks were freebies for the military if they wanted to do what they did. It was also very windy, so some of the tanks weren't always where you left them as well. So conducting these trials is very, very challenging, especially with the number of replicates that you need and the scale. You know, quite big plots, diggers, there's all sorts. There's a lot that's needed. Okay. So after the first year, the plots look how we thought they'd look. After a very wet 2017, they did not look good. And our funding was for 2 years. So we go back and we say, oh, we've put all this work, please give us some more money. One last year, and they did. They gave us one last year. And you can see that the plants really rewarded us with their extension to their life by really growing very vigorously. And that is the point at which we then harvested from the plots. So the next step, I can summarize what happened. Okay, so at the end of the trial, the aerial tissue biomass was 4.75 kilograms on those plots that contain RDX. Okay, the typical biomass for switchgrass is about twice that. So at the end of the 3 years, our plants are not nearly as big as they could have been, but they are growing in lined plots, and they've only had 3 years and a starter and a rough year to get that height up. So they could have been a lot bigger. However, our results were still pretty promising. We're looking at the aerial tissue RDX concentrations in the plants. You can see on the left-hand side, this is not detected. These are the transgenic plots. We did not detect any RDX in the leaf aerial tissues of the plants across the whole lot, all the plots that we tested, nothing. Okay. Whereas in the wild-type plots, we are seeing RDX, which harks back to that herbivory question. We were concerned that there might be a problem with our GM plants. Actually, our GM plants have got no RDX in, whereas the predators, the sheep that are on the range and other creatures that are eating the plants are already eating the RDX, so it is working its way up in the food chain. So our plants, our transgenic plants, don't have RDX in their aerial tissues. Okay, next slide. And then we looked at the levels of RDX that were in the water that were pumped off. And I think the only really take-home message here, 2018, the little purple graph there, that's the transgenic lines. We've got a lot less RDX being pumped off from the plot. And that's the kind of the crystal test. That's the champagne moment. It works. It works when the plants aren't nearly as big as they could be. And it works against all things that went wrong on the field trial due to things that were out of our control. Okay, next one. So this is kind of it. This is the way to do it. The transgenic plants removed an average of 38 grams of RDX per plot, more than the wild type, but you can argue with the wild type, there's some errors in this if there are any RDX being removed at all. It succeeded. We've gone from fundamental biology to applied science biology to field trials. We've moved from model systems into remediation relevant systems, if you like. Yes, please. And so we're squeezing through bottlenecks, but we're making it happen. And so I can say that the impact of this is the bottom line, really, is that this technology is being rolled out. They are testing it further on the US training ranges. It's GM, it's the US, they're happy with that. We did have a lot of licensing hurdles that we had to overcome as well, but it got there. And so, yeah, this is a story all the way through and something that I feel very incredibly proud of. Thank you. And so acknowledgements are necessary. I'd love to say that I did this all by my very self, like Celine Dion. Don't worry, I won't sing. But a lot of people were involved in this. Okay, I must thank, of course, first of all, Neil Bruce. I worked with Neil for many, many years on this. He was instrumental in getting this ground off the ground, started all the way through. He's been with it to the end. And everyone else who's listed there, also particularly Stuart Strand and Long Zhang at the University of Washington State, who did all the work transforming the switchgrass and the wheatgrass. And Tony Palazzo and Tim Carey, who are the guys who are actually out there physically building the plots, where I had to phone them up or email them to cajole them to drive 4 hours each way to put covers over the crops because the forecast that I saw on the internet said it was going to be very cold. Logistics, it's really quite hard, but we got there in the end, and I have something that I'm really, really proud of. And don't forget that explosives might not be foremost in your mind when you think of organic pollutants, but there's no reason why we can't apply the same approach to a lot of other organic pollutants that are out there. And I know there are very, very good scientists, many of them in the room here, that are already doing incredibly good research in that area. So I think that's finally my final slide, which is just that I wrote a little blog to discuss what went on in the field trial and things that came and went with that. That's Neil and me pushing one of the giant tanks on the training range at the time. Thank you very much for your time and your attention.
Anybody has one or two questions? Because we are already running out of time.
Okay, so we are running out of time. And if anybody has one or two questions, they can ask.
Jason.
Yeah, no, it's a good question. A question about the presence of heavy metals on the site. This was not a contaminated site. We contaminated it all by ourselves, and there were not heavy metals present. It was quite a sandy soil, so the RDX didn't bind too much. We didn't investigate too much the biological binding in the soils.
Mm-hmm. Yeah, Liz. Oh, sorry, Liz. Do you expect a similar effect when you have aging of the RDX in the soil? Because you spike, but when you have the aging of the RDX, do you expect a similar mechanism for phytoremediation?
We do with the RDX because it does not bind tightly to the soil, but it's a really important point you make that you need to age the soil with your pollutant because then different chemical and physical things happen to the soil and the pollutant. Yeah.
Yeah, being organic compound, how is it taken by plants?
TNT and RDX, they're very lipophilic, and so they are passing through the membranes by passive transport, whereas metals, for example, have specific transporters. So yes, they just go straight across the membranes.
So just one more minute because we were not able to take the photographs for the Milton Gordon Award, and the society needs the photographs. So we will be redoing again for the award ceremony, and Jason will do the honor again. Thank you. Thank you.
Thank you, ma'am. Now, may I invite Professor Om Prakash Dhankar to introduce the keynote speaker. Sir, please.
Okay, it is my honor and distinct pleasure to invite my close friend and one of the distinguished professor, Varaprasad. He is a distinguished professor at the University of, distinguished professor, as well as the Krauss Endowed Professor and Director of the Feed the Future Climate Resilient Sustainable Intensification Lab at the Kansas State University. Dr. Varaprasad has he obtained his PhD from the University of Reading in UK, and also his research is focused more on understanding the responses of crops to changing environment and developing best management strategies to improve the project yield. Vara has published almost close to 500 publications, and he's a very high index, H-index and all those, and he's one of the top highly cited researchers in the field. Also, as I know Vara is a close friend, we interact a lot. I know he's down to earth, but he hardly kind of like highlight his achievements, but I can tell you he has got almost more than $200 million for his research funding, and currently, just last month, he got another $100 million award from the U.S. Agency for International Development for capacity building in the different developing countries in Africa and Nepal and Cambodia and other countries. So with that, I will invite Dr. Vara Prasad to deliver the keynote lecture.
Yes. Good morning, everyone. Thank you very much, Om, for your kind introduction. I know we are running about 15 minutes late, so I'll try to cut down my shot by at least 5 minutes or more. So I'll get my timer started here. So first of all, thank you very much for giving me this fantastic opportunity to speak to you today. My special thanks to Jose Puttur and Om Prakash for giving this opportunity. And I thought that my title of my presentation, I put it as Phytotechnologies for Food Security and Environmental Sustainability. I know a lot of you will be also talking about the food safety issues. So I'm going to not talk about food safety, but restrict my talk to food security and environmental sustainability. Looks like, let's see if this moves. Okay, let's try it. Okay, tap it. Okay, it looks like there's about 3 or 4 seconds lag, so I'll try to move the slides a little bit ahead of time. So we have many global challenges with us. Number one is about food insecurity. About 828 million people do not have access to food. And we also have issues with nutrition security. About 2 billion people are malnourished, both undernourished and also overnourished. And we have issues with climate security. Earth has warmed up by 1.2 degrees Celsius compared to beginning of the last century. If we go business as usual, we're going to warm up again by more than 2 degrees Celsius. We have issues with water security. Prolonged drought and limited water is an issue. And in some places, actually more water or high intensity rainfall is also an issue. And we have energy security issues, and more importantly, we are dealing with equity and inequalities for access to food, resources, knowledge, and they're not equally distributed. There was a report last year about the current status of the Sustainable Development Goals. We all know there are 17 Sustainable Development Goals. We move forward with the Millennium Development Goals. We didn't meet those demands, and then we set up the new ones. Unfortunately, only 15% of those were achieved last year, which was midway through the point where we thought that these will be met by 2030. We are already halfway through, and only 15% of them are on track. So that's not a significant progress, but hopefully we are able to make that changes. But when you look at the food production, majority of the food grain crops shown here on your right-hand side, maize, rice, wheat, and soybean, we have made significant increases in the productivity. But on your right is a relative yield gains. Those relative yield gains have been very low. In the initial years, they were always more than 2 to 3% a year, but now we are less than 1%. And to meet that 60% increase in the food production, these numbers have to go back up to about 2% per year to meet those goals. And I would like to now talk a little bit about agri-food systems and phytotechnologies. When we talk about either agri-food systems or phytotechnologies, we should take an end-to-end approach, meaning that we should start from the production, distribution, storage, packaging, marketing, consumer preferences, preparation, and waste recycling. So the entire cycle needs to be taken into consideration, whether it is agri-food systems or also when you're developing these phytotechnologies, we really need to be thinking about the end-to-end systems so that we can work together. And then this report came out last year or earlier this year, the global agri-food systems, the hidden cost of those. So on these visuals show that the total hidden cost of our agri-food systems is about $12.7 trillion. And about 73% of that is related to health. That is related to diet, that is related to food. And only 22% of that is environmental, and 4% is social. So, and then of those environment, nitrogen, climate, and land had the major issues. And then both developed and developing nations were in the equal split. But in developing nations, it was more about environment and social issues, not having access to food. Whereas in the developed nation, it was the health issues that needs to be addressed. So to consider this agri-food system and to take care of those, we really need to be thinking about transdisciplinary research. And if you look at the environmental cost in terms of greenhouse gas emissions of global agri-food systems, 26% of the greenhouse gases come from agriculture. And then about 50% of the world habitable land is in agriculture, 70% of the freshwater withdrawals, and also causes eutrophication as well. So phytotechnology should be focused on minimizing or decreasing these greenhouse gas footprints. And that leads to me the actual, what are the platforms available so that we can address these global agri-food systems in terms of greenhouse gas emissions or climate smart. So I would like to propose 3 of these. One is called circular bioeconomy systems. Second one is broader One Health. And third one is sustainable agricultural intensification. And we need to be innovative in our phytotechnologies in our agri-food systems so that our net negative greenhouse gas emissions, especially we can sequester more carbon. Agriculture is the only field which can actually sequester more carbon than we produce. Any other sector, we can maybe become neutral, but we cannot really sequester more than what we produce. Agriculture has that potential opportunity to deal with that situation. My slide. Okay. So circular bioeconomy, first principle. Here is the goal is the production, utilization, and conservation and regeneration of biological resources with emphasis on renewable and minimizing waste. We have a lot of inputs in agriculture, and we have a lot of low input use efficiency, whether it is seeds, water, nutrients, carbon, energy, labor. And then so that requires the whole system redesigned to improve efficiency and recycle and manage our waste. We all know about the 5R principles for water and nutrients and energy: right source, right rate, right place, and right time and right manner. But when it comes to waste management, there are 6 R's: refuse, reduce, reuse, recycle, repurpose, and residue management, so that we can have a closed loop in that system. And that closed loop could be applied to water, nitrogen, methane, carbon, and more as well. So that's the circular bioeconomy system, where innovations in phytotechnologies will play a significant role. Second platform is broader One Health. We know about One Health, but One Health right now only considers 3 components, which is animal health, human health, and environmental health. But it misses the 2 most important components where agriculture or food systems begin, which is soil health and the plant health. So I'm a big proponent of broader One Health to include that. Soil health and plant health is included. If you have healthy soils, they'll lead to healthy plants to healthy animals to healthy humans. And then if you have all of those, you'll have healthy ecosystems. So we are missing the 2 most important pieces. So I would encourage all of you to be thinking of broader One Health rather than just focusing on animal, humans, and environmental health. And that also requires systems and transdisciplinary research where biophysical scientists and social scientists should come together to address these challenges. And the last platform I'm going to talk about is sustainable intensification. So the goal here is to provide access to safe, nutritious, and healthy food at all times to all people from existing farmland without damaging our ecosystem health. But unfortunately, considering what has been happening, without damaging is not going to solve the issues, because we have already caused so much of damage that it needs to be rebuilding, or regenerative agriculture is the key. Regenerative agriculture is the key. So without damaging or do no harm is not going to be enough anymore. So there are multiple components to it: genetic intensification, which is higher yields, resistance to pests and diseases, tolerant to abiotic stresses, improved nutrition, medicinal value, improved health. And then we have the second, agroecological intensification, which is more about farming systems, improved soil and water management, pest management, weed management, efficient agricultural practices. That covers the agroecological intensification. And third one is the socioeconomic intensification. It is not just enough to produce, but we need to have a market to sell at an appropriate price in that market so that farmers can actually profit from their agriculture. So developing markets, creating new markets, and also understanding the barriers of adoption, because a technology is not a technology if it is not adopted. So we need to be thinking about adoption and scaling a lot. So creating enabling environment and institutional building are part of it. So basically, and then all the new tools, the geospatial, artificial intelligence, nanotechnology, mechanization, precision agriculture, private sector engagement are all integral part of these systems. And then basically what this gives you is a G by E by M by S interaction, which is genotype by environment, by management, by social interaction. So that's very critical. And this will also require transdisciplinary research and our phytotechnologies, whether any of these 3 components or 4, the social piece is also very important important, which needs to be addressed. So now I'll give you an overview of what my Sustainable Intensification Innovation Lab is currently doing and highlight some of the specific phytotechnologies which we are using, not only to improve the productivity of the major crops, but also take care of our environment at the same time. We are currently active in 14 different countries. In Asia, we are in Bangladesh and Cambodia. In East Africa, we are in Ethiopia, Tanzania, and Malawi. West Africa is Burkina, Niger, Mali, Senegal, and Ghana. And Latin America and Caribbean is Honduras, Haiti, and Guatemala. So we have multiple projects. It was the first phase of this was from 2014 to 2024. It was a $75 million grant, and I'm very pleased to inform you, as Mo mentioned, that we recompeted for this grant and we obtained it again. So it's going to be another $50 million for 5 years. And if we do a better job, maybe we'll get another $50 million. So that's a $100 million grant. So through this, we have multiple, in each of those countries which I mentioned, we have research programs which is focused on farming systems. We have a mechanization consortium. We have a geospatial consortium. We have a spatial consortium. Policy consortium, we have a soils consortium, and we have 4 centers of excellence which serve these 4 major regions of the world. One in Cambodia, one in Senegal, one in Haiti, and one in Guatemala. So there's it's a lot of people involved in it. So it's a team effort. We have about 130 scientists, more than 120 organizations, including 6 US universities, 9 CGIAR centers, and we have provided funding to more than 250 master's and PhD students from those 14 different countries. So it's truly a multidisciplinary and multi-institutional partnerships. So this is what I was going to say, that our new innovation lab we renamed as a Climate Resilient Sustainable Intensification Innovation Lab. So the climate resilient piece is new, which involves both adaptation, mitigation, and resilience. It started last month. There'll be official announcement of this will happen in the World Food Prize event in the next week or so. But we were told that embargo has been lifted. You can share that information. That's why I'm sharing it. We have 8 target countries: Cambodia, Bangladesh, Ethiopia, Tanzania, Senegal, Ghana, Guatemala, and Honduras. So the mission is to innovate and find evidence-based, practical, accessible, and scalable solutions to the challenges of our agri-food systems. The key thing here is scalable solutions. And then the goal is to empower people and institutions with knowledge and innovations that positively impacts agriculture productivity, environment, and natural resources. That's going to be the focus of it. So I'm already 10 minutes into my presentation. So I have 18 minutes for the 30-minute mark. So what I'll do is that I'll cover 1 minute, 1 technologies. So I have 17 technologies, and hopefully I'll be able to cover them. So if anyone needs more information or wanted to dig a little bit deeper, I'll be here all day today. So please stop me at the lunchtime or tea time. No matter where I am, I'll try and answer those questions. So here we go. 18 minutes, 17 technologies, and 1 minute for summary. The first one I would like to talk about is speed breeding. The traditional breeding technologies takes about 8 to 12 years for the release of a variety. And we all know that much time, whether it is a disease or an insect, the insect and the disease gains resistance. So by the time you release your variety, the pest has already gained resistance. And so we have now speed breeding where we can have 6 generations in a year, where you control the lights and the photoperiod so that you're hastening the life cycle. So that's one new technology which is available now and has a global implication. And the second one is when you combine this speed breeding with your CRISPR-Cas technology, CRISPR-Cas9, that is genome editing tool. So you are not only fastening the genetics, but also the generations. So both genetics and breeding you are increasing, and that's what they call it. They are calling it an express edit plus speed breeding, and that's also a new innovation. So by doing this, you are not only able to genetically modify, but also breed faster with your breeding tools together with that. So that's number 2 innovation. And number 3 innovation is stress-tolerant crops. Every crop has a different cardinal temperatures, meaning different optimal and ceiling temperatures. And here, this is my almost 10 years worth of my work. My 5 years of my couple of years of my PhD and couple of years of postdoc. So this summarizes significant amount of my early career work. I developed temperature response curves for all of the food crops. I'm only showing you a few of them here. So here's wheat, rice, sorghum, and millet. So the ceiling temperature, meaning the point at which that does not produce any more grain, which is harvest index on your y-axis. So millet, pearl millet, is the most heat-tolerant crop, followed by sorghum, followed by rice, and followed by wheat in terms of cereals. When you look at legumes, soybean is the most tolerant one, but mung bean and cowpeas are even more tolerant than those two. So you have different crop species with different susceptibility or tolerance to temperature. So we need to identify those crops and put those into those ecosystems. And every one of us knows here, the globe knows that last year we celebrated the International Year of Millets. So millets is not one crop, it's a family of crops. The major millets being jowar, bajra, and ragi, which is sorghum, pearl millet, and finger millet. And we have other millets, minor millets, and many more of those. There's a lot of genetic diversity in these millets. And millets are key for food, nutrition, climate, and water security, because as I told you, they are not only they're the most heat tolerant. And if you look at the water requirements of those, when you compare rice is about 2,000, when the millets are less than 250 or 300 millimeters of water to complete their life cycle. So we need to be capturing, using those crops which are climate resilient in those technologies. And not only those crops, there is a lot of genetic diversity and a lot of varieties differ in their stress tolerance. I'm just giving you here an example of a program called DTMA maize, which is drought tolerant maize for Africa, later on renamed as stress tolerant maize for Africa. This was funded by USAID and Bill and Melinda Gates Foundation, and supported by IITA and CIMETS. They've produced about 184 distinct varieties. These varieties produce more than 50% yield, and then in some areas under drought stress environment, we actually double the yields. And then more importantly, there was no penalty under the normal conditions. So these type of varieties are available for other crop species as well. That needs to be explored. And we should be also looking at native and indigenous species which have more tolerance and more nutritional value. Here is an example from Tunisia, which is a species called Sula. And it's a forest species, and it's extremely useful for land restoration. So again, for pasture land restoration, we have such species of plants which can be used and should be used. So these are just one or two examples, but I'm sure that your indigenous Kerala, there's plenty of those species which can be utilized. And then when you talk about climate resilience, we need to be thinking about conservation agriculture, the three principles: minimum tillage, continuous soil cover, and crop diversification. And with minimum tillage, we sometimes do not see the yield benefits right away. Sometimes in the initial years, you may have actually yield losses, but after using that minimum tillage or no-till for a few years, you actually gain carbon and organic matter buildup, and then you see better benefits. But again, we are already talking about these technologies, but we are not breeding the varieties to be responsive to minimum tillage. So the focus has to be for identify those varieties which can withstand this minimum tillage, meaning that they have early emergence vigor, deeper rooting systems. So those needs to be identified as well. And then the eighth technology is related to vegetable crops. And then there's a lot of opportunities, especially when we talk about engaging women in agriculture. Women are already taking the lead in agriculture, and they are the people who actually provide nutrition and also grow the vegetables which are very nutritious. So we were working of indigenous or commercial vegetable crops which are more responsive to conservation agriculture principles. So here are some examples from Ethiopia, where we are growing indigenous crops like garlic, potato, cabbage, indigenous varieties of those. So we are asking women farmers to use whatever the locally available mulch is, and then locally available mulch for providing continuous soil cover. And we are using drip irrigation system. And on top of that drip irrigation system is supported by solar power. And we're also using solar power to lift the water, because women go miles and miles to fetch water to plant, to feed their plants, and also use it for consumption for their own families. So we are talking about water lifting technology, solar power, and also water delivering system using solar power. So this has been very successful in Ethiopia, and we are replicating this in multiple countries in Asia and Africa as well. So now coming to technology number 9, this is responsive to intercropping, because our commercial crops in US and elsewhere, we grow crop rotations. We grow we go single crop in a year, and then following that we go another crop that is called as crop rotation, which obviously is very important and valuable. But in smallholder farmers, for the climate resilience, or I should say just resilience, they do always grow multiple crops, and sometimes multiple crop species are put in the same field of land, and intercropping is very common. So again, we need to be thinking, we have seen that at least 20% yield increase when you do intercropping in many areas when compared to that continuous monocropping. And again, we need to be thinking about varieties who are suitable for these intercropping systems, because they need to compete for their solar radiation, but at the same time, they also need to compete for the roots and also nutrients and water. So we need to be thinking about that. And number 10 technology is doubled-up legume system. When we talk about intercropping, you commonly see that you have a cereal, which is either your maize, sorghum, pearl millet, and you have your legumes, which is either common bean, groundnut, or cowpeas, right? So when you do intercropping, we do only 2 of these crops. But this is called doubled-up legume system, where we are introducing a 3rd legume, which is called pigeon peas. So you have maize, we have pigeon peas, and underneath the canopy of the pigeon pea, because pigeon pea is much taller, underneath the canopy of the pigeon pea, you can still produce your groundnut or cowpea. So that's what is called a doubled-up legume system. It improves the efficiency, it provides more yield, provides more market value, and that adds more profits to the farmers as well. Number 11 technology is dual-purpose crops, because most of the time farmers not just grow single crop, they grow multiple crops. Not just multiple crops, there's always some component of a livestock, whether it is cattle, whether it is goats or sheep, or whether it is chickens, or whether it is aquaculture fish. So there's always multiple, it's a farming system. So we are looking at crops where grain is used for human consumption and the biomass is for the cattle, because sometimes actually farmers make more money by selling the biomass in the off season than the grain itself. So when you give them high grain yielding varieties, they're not adopting it because they need the biomass as well. So these are called dual purpose crops, meaning grain for human consumption, biomass for livestock, and both grain and biomass have high zinc and high iron in them. So that has been very popular. We have those varieties for sorghum, pearl millet, and cowpea, and which are becoming very popular in West Africa. Number 12 technology is agroforestry. When we talk about farming systems, especially in the semi-arid region or semi-arid tropical regions, there's always a vast land. Trees are an integral part of those ecosystem. And here is an example of agroforestry system, which is acacia tree. This is a peak growing season. The tree is naked, whereas underneath the tree, the crops are very well done. So if you go in the off season, the tree will be full of flush of leaves, and there will not be any crops. So it's not competing with resources. It's called reverse phenology, meaning the trees and the crops are growing in a different timeline. And the second phenomenon is hydraulic lift. These trees have a deeper root system. They bring the soil, water, and the nutrients above to the above level, and that will be available for the farming systems, for the crops. And then in the hot times, off season, these trees are lush green, so cattle come and sit underneath them. So that provides additional organic matter. So it's a multiple system. And we also have a similar shrub system with Anguria senegalensis and Peleostigma. So number 13 technology is inclusion of perennials. So I talked about agroforestry systems, but in the vegetable system, especially for the women farmers at the homestead, they grow these commercial vegetable gardens, but the borders are left empty. So you can actually grow some perennial trees on the borders. And one example here is from Cambodia is Acacia pinnata, which is grown on the borders, which has very high vitamin A and also used for human consumption and also for livestock. Another example, very common example, probably many of you know, is moringa, which is also has the leaves have a value, pods have a value, and the entire tree is extremely valuable, and it's a perennial as well. And on the right side is a wild gardens, where you have multiple species combined together so that you get year-round supply of some sort of nutritious fruits so that you can feed your family, and you can also sell them to get additional markets. So that's number 13 technology. So I think I'm doing okay on the time. I have 5 minutes and less than 3 technologies, so I'm doing well. So the fourth one is I talked about the perennial agroforestry systems, and I've also talked about the perennial shrub systems, and I also talked about perennial border systems, but the new future is going to be perennial grains. I know some of you may not like it. We have took 100 years to convert many of these perennials into annuals, and we are actually turning back and going the other direction, but there is certainly some value to that. And this perennial grain crops, we have 2022 science or the AAAS, one of the 10 scientific innovations or breakthroughs was perennial rice, which was originally started the work in Land Institute in Kansas and Salina, and then later on in collaboration with Chinese Academy of Agriculture Science and University of Queensland, they released the first perennial rice, which is 8 cuts, same yield. 8 cuts, same yield. And of course, there are the availability and other issues that are still being worked out. Logistics, because those varieties are not available for the farmers in West Africa or East Africa or in Asia. So hopefully those IP issues will be resolved some point in time. Otherwise, there is still opportunity for breeding for these perennial crops, and work has been in progress for perennial wheat, which is a Kunza example at Salina. Kansas, and there's also a perennial sorghum work because when you use perennial grains, there is a lot of root biomass which goes, and then a lot of energy savings and also labor savings goes into that. And then now moving a little bit beyond those production technologies into pest management technologies. This is integrated pest management. ICPA is an international center for insect physiology and ecology in Kenya. They're doing this work. It's called push and pull technologies, meaning that it's a trap crop basically. Here is an example of a stem borer in corn or maize. It's a very prominent insect which damages. So what they do is they intercrop with Desmodium. Desmodium is a legume. They intercrop this Desmodium into the maize, which releases volatiles, which repels the stem borer from landing onto your maize crop. And on the borders, they produce Napier grass or Bracharia grass, which pulls this stem borers away from your main crop. So that's why it's called push and pull technologies. And I'm sure there is a lot of indigenous species in India and elsewhere which can be used for a similar push and pull technology to combat your insect pests. So that's that. And the second last technology is biofortified crops. I did give you an example of the dual purpose crops which we are working on, which are high in iron and zinc. But this is a Harvest Plus program which ran for many years, 15 or 20 years, also got the World Food Prize. And they have biofortified crops and varieties which have been released, many of them, many crops, but the more important ones are high iron. You have high iron beans, cowpeas, lentils, and pearl millet, because anemia is a major issue, zinc deficiency is a major issue, and then vitamin A deficiency is a major issue for humans. So you have this high iron beans, cowpea, lentils. You have high zinc wheat, rice, maize, and sorghum. And for high vitamin A, you have orange flesh, sweet potato, cassava, maize, banana, and plantain. And I want to emphasize that these are all non-GMO. These are used by traditional breeding practices, so there is nothing to worry about, or it doesn't come into those categories. So this has been widely adopted. And the last technology, with 2.5 minutes to go, is going to be bioremediation. And this entire conference, I'm sure you're going to hear a lot of those, whether it is phytoremediation, whether it is rhizoremediation, or it is bioremediation, which combines all together. So I'm not going to dwell upon this particular technologies. And this is the award winner a few minutes ago gave a fantastic presentation of RDX and TNT, but we have a lot more arsenic, mercury, and I can go on and on. So the phytotechnologies are available to not only remove from the soil, but more importantly, after these plants remove it from the soil, where are you going to put that plant? So you need to be thinking about how do you dispose these plants, because now these plants have concentrated amount of these pollutants, so you have to make sure that they are disposed appropriately. And I'm sure there's a lot of microbial cells, another big thing. So it's a microbiome, soil microbiome, plant microbiome, how those play a significant role in detoxifying our soils, and then using an appropriate disposal of those things. So we have a lot of opportunities. So with 1.5 minutes to go, I would like to conclude. Hopefully it was a rapid fire. I'm sorry it went a little faster. But as I said, if you need more details about any of these technologies, please do let me know. And I gave the the people who developed the technologies and the institutions which developed it and which we collaborated with all was given in each of those slides, and along with the name of the scientists who worked on it. So I would encourage you to either contact me or you can also directly contact with them. So to conclude, agri-food systems are complex and have many challenges and wicked problems, and solutions require systems thinking and convergence of science. I'm not only talking about convergence of biophysical sciences, but also convergence of social sciences in it. And our phytotechnologies and innovations should be productive, profitable, resilient, resource smart, nutrition smart, and climate smart. And focus should be on more nutritious, safe, and healthy foods, and not just do no harm, but actually rebuild your natural resources. And that's what our innovation lab will be working for the next 10 years. And there are opportunities to make our agri-food systems to be net negative emissions with emphasis on broader one health, with healthy soil, healthy plants, healthy animals, healthy human ecosystems, humans and healthy ecosystems. And then using principles of these sustainable agricultural intensification, broader one health, and also circular bioeconomy systems, we should be making sure that our phytotechnologies are aligning with them. And lastly, transdisciplinary and convergence of biophysical engineering, health, nutrition, and social sciences is essential and key to success for developing, adoption, and scaling new innovations in new innovative technologies and innovations. With that, thank you so much for giving me this precious opportunity to speak to you today. Thank you very much. Okay, looks like he's going to give me a little bit more time to answer your questions, maybe.
Thank you, sir, for the valuable and insightful contribution. Now there is a time for discussion. You can ask 1 or 2 questions.
Fantastic. Yeah, lots of students. So there's, go ahead. Yeah. I think for the students, I think you are in the right place to be focusing on agriculture, because agriculture is food, agriculture is nutrition, agriculture is health, agriculture is business. So that's what you need to be thinking about. So you are in a very good spot to be building your careers with a lot of innovations and technologies happening. Jason.
Beautiful presentation. So all of the technologies you presented, they're all by traditional breeding practices?
Yes.
A couple of those, I know that there's work on amendments, soil amendments that will either produce, equivalent benefits or maybe benefits on top of traditional breeding. Is there interest in that type of approach in these programs?
Yes, absolutely. Because what I presented you was more on the plant side of it. But I think as long as the impacts are additive, meaning anything which you are going to add to soil or plant, if they are additive in nature, that's absolutely important. What do I mean by additive is that there's no negative interaction, that positive things with additional inputs, if they're negative, then you level out. So as long as any innovations you're adding on top of the existing one, if the effects are additive, that's going to be fantastic.
Very interesting, highly interesting. My small question is, you said perennial rice, you can have 8 cuts, same yield.
Yeah.
Is it now in practice in the farmer's field?
Yeah.
The second part is, if it is in practice, can it be expanded to other countries where rice is a major crop?
Yes, so that was the innovation of the year from the AAAS a few years ago, and right now it is being in practice only in China, in Hunan province, because they're still testing it on the farmer's field. The results are from the farmer's field, and they have an IP. I think it's the number is 23. I think PR 23, or don't quote me on that, but that's a line which is currently being used, and that's a cross between the wild species and the regular species of rice. So I believe it's going to take another 5 to 10 years before this will be open for other countries. Right now it's currently being only grown in China and elsewhere. So what we have done, considering the geopolitics around the world and our focus in West Africa and Africa, so we are encouraging the local rice institutes to initiate their own program, because we know the species where the gene came from. We have a genome sequenced. So if somebody else wants to restart it and actually put in their own rice varieties, because this was done in, this gene was introduced in their rice lines. So if we can do in our own rice lines, it will be great. So I'm proposing that, or actually looking into digging a little bit more deeper into perennial rice systems. And of course, perennial sorghum is also there, which has significant opportunities. Yes, sir. Yes, 8 harvests. Great point. So I would admit that so far the results are saying that it's either relatively low. I would stop there, because our more research needs to be done, because the very short time, because you're not, because nutrients plant has to get it from somewhere, right? So that has to be provided to them, and especially micronutrients. So I think adding some other technologies on top of that, so that you can provide these micronutrients, whether by foliar application or whether your soil amendments. So those needs to be added on this, provided we're not focusing only on nitrogen, phosphorus, and potash. So that's the piece which needs to be taken care of. Thank you. Yes.
Beautiful presentation, Professor Bala Prasad. Just an extension of what Professor Sangwan asked. Mm-hmm. What is the productivity versus the nutritional content and versus the disease and pest resistance of the perennial rice?
Yes. So right now, because it's a system, right? Perennial rice is a system. So right now, the yield levels are okay. Labor savings are okay. Input use efficiency is high. Input use itself is high. The issues we have to deal with is nutrition and also their impact on the pests and diseases. Because in the normal system you have rotations. If you're monocropping, you're managing a lot of insecticide and pesticide, and you always have a rotation. But if you're growing one crop for 5 years or 4 years, and again you have to break the pest cycle. It's encouraged that 5 years, 8 cuts or whatever that is, and then later on break it again for 2 years or 3 years and come back to that. When I say perennial, it's not like it's not like a tree system where you're going to grow it for 10, 15 years, but it's just about 5 years, break it to break the pest cycles and weeds. Let's not forget about the weeds. That's very critical. So pests, weeds, and then nutritional value still needs to be figured out, and that can be overcome by additional different phytotechnologies, trap cropping, push and pull technologies, amendments, soil amendments, plant amendments. So there is a lot of opportunity. Let's not restrict ourselves saying that if it is lowering yields, but again, you have this, you also saw the Harvest Plus, right? So there could be, maybe those genes should be incorporated into high zinc rice, high iron rice, or high vitamin A rice, so that you boost the nutritional value of those as well. Again, it's a working the entire system, not just focusing on one particular crop, because farmers never grow one single crop. They always have multiple crops, multiple livestock system, so that's a part of their resiliency, whether it is for climate or whether it is for market. Thank you.
Well, I know there are so many questions more, but keeping in view of the time shortage, we have to cut the discussion short. But you can interact with both plenary speakers during the lunch and during the conference time. And so both speakers did an excellent job, set the stage for excellent discussion and science for the next 3 days. Please join me, give a big round of applause for both speakers for excellent job. Thank you. And I will invite Professor Josh Puthur for a small token of appreciation as well, and some announcement.
I invite our senior professor from the department, Professor Santosh Nambi, to come up to hand over our small token of love. Quickly, I will give you a few tips that will help you in the further sessions. Don't go away. We have photo session. Be around. I think after tea, we have the photo session. And that will be in the physical education block. We have planned it. So anyway, we will guide you, be around. Then another thing is that after this, we have 2 parallel sessions going on. From the schedule given to you, you can get it what is going on over here and what is taking place in the physical education. So to physical education block, you have to walk through our food court, go a little down, and it is the same building which is behind our food court. And there are always, signs are always kept over there, and you can go to the seminar hall of the physical education block. During this lunch hour, the poster session starts. So kindly hang your posters, but we have the evaluation and visit tomorrow. In between, if you want, you can visit anytime. You are welcome. And when you see this button badge, this is the guys from our department. You can always, our guests can take help from them. Any help you can go to them. They are very hospitable. They love to help you. And this side we have kept some black tea, lemon tea and all. If in between you want to go, you can prepare yourself, have lemon tea and black tea. Also water is there. Go this way, get it and help yourself. Thank you.
You can announce maybe we start just 10 minutes late at the sessions.
Yeah, we are late running, but we won't compromise with the schedule. We will be on time, but he has given a relaxation of 10 minutes. So 1:40, we are starting our next session. Please keep up the timing. Thank you.
Kindly note, 3 of the presenters have not given their presentation to the presentation desk. Kindly provide your presentation. Dr. Elena Maestri, Professor Rajendra S. Sangwan, and Sanika Jain. Kindly provide your presentation to the presentation desk. Presentation counter is just next to the registration counter. So kindly provide your presentation to the registration desk.
Maybe we can start. Good afternoon, everybody. I am one chair, Elena Maestri, and here's the other chair.
Rana Pratap Singh.
Didn't want to mispronounce it. So this session is on metals and metalloids. We have 5 presentations. Most of them will last 15 minutes for students. And the 2 students are under evaluation. I will read you briefly some instruction just so you know. Okay, the presentation time will be very strictly enforced. There will be presentations of 20 minutes for the postdoc scientists and 15 minutes for the presentation. In all cases, we have to leave 3 minutes for the discussion. And the discussion, please use the microphone because they have to be transmitted online. There will be a warning bell 5 minutes before the end of each presentation, and the final bell more evident 2 minutes later. So we will leave 3 minutes for the discussion. And the student competition for the best oral presentations, we have today 2 students, Sanika Jain and Jose Angel. They will be evaluated by anonymous reviewers in the audience, and then we will have at the end the results. The first place will be $200 with a certificate, a medal, and a gift pack. The second place, $100 and all the rest. The third place, $50 and all the rest.
Only 2 are there.
Okay. So, shall we start?
Yeah.
I move.
So the first presenter in the session is Professor Elena Messi, co-chair of the session, and she is from University of Parma, Italy. She will speak on biofunctionalized biochar to alleviate the effect of cadmium and arsenic in Solanum lycopersicum. So I invite Professor Elena to present her work before the audience.
Okay, thank you and welcome. I'm here to give you a presentation about a new project that we have just started, so the results are very preliminary. And the title speaks about biochar and biofunctionalization of biochar, together with the alleviation of effects of metals and metalloids in Solanum lycopersicum, which is tomato. It is interesting to highlight that this project is a cooperation between Italian institutions and Chinese institutions financed by our ministry, which encourages this kind of international cooperation. So the Chinese partner is Guangdong University, represented by Xuanjing Ma, and the other authors, myself, Marta Marmiroli, Nelson Marmiroli, and Luca Pagano, we belong to the University of Parma, but organized in a consortium, which is Consorzio Ital Biotech, is a consortium of different universities and Italian companies, which deals with biotechnology. So this project, which is called FunChars, the acronym stems from the words functionalized and biochars, because this is what our project will deal with, but focused on soil remediation strategy, essentially for metals and metalloids. We want to understand how the biochar, which it has not yet been mentioned here, but you probably know it is a carbon compound derived by pyrolysis or pyrogasification of biomass. So this biochar, which can be a method to valorize biomass in different contexts, can be functionalized, so with some properties added from the chemical and biological point of view, which can become a strategy, an energy efficient strategy, because it will come from a pyrolysis process, but together with the purpose of helping in the contamination. So the idea is to enhance the properties of biochar, in this particular case using microbial inoculation, not yet chemical compounds. And we want to functionalize the surface of the biochar also with nanomaterials. This will maybe be explained in the next Phyto conference. The idea is that the microorganisms added to the biochar and the biochar used as amendment in the process of phytoremediation will interact with the rhizosphere of the plants if we choose specific plants which can be suitable also for phytoremediation. We want to study in this project the interplay and the kind of mechanisms that can intervene between the biochar process, the biochar mediated process, and the plant-based process together with the intervention of microorganisms. And the idea of the cooperation between Italy and China is also to understand some socioeconomic and legal aspects of this research, which of course will be very different in the two countries. So the main objectives of this project will be, first of all, to develop novel solutions for remediation, starting from the idea of utilizing functionalized biochar. Then to try to implement or suggest the strategies to implement this kind of approach to increase crop production, especially in soils which might be contaminated. We heard about it also this morning in the plenary. In some way, helping to increase food security by producing crops also in slightly contaminated environment. At the same time, valorize or reutilize biomasses which come from the normal agronomic practices. All this should lead to design some alternative strategies, also considering how we measure the impact and the effects of these combined biochar and plant solutions, in order, at the very end, to provide a sort of support in decision-making and understand the applicability of these instruments and strategies. We have to remember that biochar per se is a very rich carbon material, which in any case assists in carbon sequestration, so it is considered a carbon negative or carbon neutral technology. So at the same time as working as an amendment in soil, it can also improve the, let's say, the counteraction towards carbon emissions. So the rationale was, in these first experiments, it was to choose systems that we already knew in our laboratory, so the combination between tomato plants and some specific bacteria, Pseudomonas fluorescens and Azotobacter crococcum, that were already utilized in the past, together with biochar derived from residual biomasses from the agriculture, so to keep everything in the same loop and work together. The biochar, in this case, came from a mixture of residual biomass. We are studying it in different projects, so let's say it has been chosen for the purposes of another project on circular economy, but we also wanted to apply it here. The residual biomasses are, let's say, mixed, but the main part is biomass deriving from the sunflower cultivation, which, by the way, due to climate change, is moving up, and it is also in our area. We are in the north of Italy. Previously, it was very rare to see sunflower, but now we have also sunflower in our regions. The characterization of biochar usually involves a morphological I don't know how to use the pointer, but you can see it here. But the morphological evaluation with different methods, including transmission or scanning electron microscopy, and a panel of chemical and biological evaluations. In this case, we try to follow very carefully standard methodologies, which are in Europe and in Italy. They are, let's say, the same methodologies, standard methodologies that can be used to characterize the fertilizers, amendments, and this kind of products. So for measuring the pH, the conductivity, the density, and it is a fact that biochar is almost always high pH. This one is about 9.5 more or less. The conductivity is a measure of the quantity of salts that you can have in the biochar, and it can be a discriminating factor in some cases. The density, or better still, the granulometry, is very important because the morphology of the biochar tells you how you can use it and distribute it more or less easily, and there is a distribution, let's say, of granulometry class. Basically, the biochar retains the granulometry of the starting material, but it can also be modified later on. Then you can measure the content of organic matter and ashes, humidity, and, which is important in our case, the content of metals, because when you use pyrolysis on a biomass and some of the materials can be concentrated during the process of pyrolysis, some of the metals could even reach above the limit which is allowable, let's say. We don't have a specific legislation about metals in biochar, but there are some guidelines that you have to follow, so we always check the metal content. And we also check, which is not in this table here, from the biological point of view, if they stimulate, if the biochar stimulates the growth or if inhibits the growth. In this case, it was considered a stimulant of plant growth. So we start from a biochar which is already per se a good starting point. Then we choose these two bacterial strains based on our previous experiences because they were part of previous projects financed by the European Union, in which we combine them with other kind of bacteria, but we choose these two specific one, Pseudomonas fluorescens and Azotobacter chrorococcum. They both act as plant growth promoting microorganisms, but with different impacts, because Azotobacter, the name says it all, is involved in nitrogen fixation, so it can contribute to the fertility of the soil. But interestingly, it has also been used already in combination with plants for bioremediation purposes. And Pseudomonas fluorescens, specific strain, because there are many different strains, is works in biocontrol of pathogens, which in tomato does not, it is not a bad thing, and possibly also phosphate solubilization. The idea of plant growth promoting microorganisms is that they interact with plants, they interact with the rhizosphere, they produce stimulants, they receive carbon sources from the plant, and altogether they improve the nutrient, let's say, the mineral nutrient of the plants, but also they can exert effect like, for instance, biocontrol of pathogens. So there is and Jason was mentioning it this morning, amendments and biostimulants, they are now very interesting for the use in agriculture. This is the previous experience I was telling you about. We have worked with tomato, wheat, and maize using a microbial consortia developed in the SIMBA project financed by the European Union, and among these bacteria we had also the two that I am talking about. We have developed, or we are still developing, real-time PCR methods to trace these microbes in the rhizosphere even after the application, and we have made some studies in wheat and tomato fertilized with this kind of microorganisms, with or without biochar, to see how they impact on the quality of the products and the performance of the plant. So we had already a good material to start, and this is why we choose this specific combination, tomato and microbes. Biochar functionalization is not something that has a protocol, let's say, so everybody more or less tries to do it their own way. In our case, we grew the bacteria and we simply incubated bacteria and biochar together in a constant rotation for 24 hours. So the biochar became, let's say, impregnated in the micropores and the micro niche of the surface. It should become impregnated with these microbes. The first test that we have performed was just to compare the behavior of plants without any amendment, with biochar alone at 2 doses, and the 2 doses of biochar with microbes. So basically a standard comparison of the effect of biochar functionalized or not. And the results with tomato, of course, the first test was in a greenhouse. They showed a limited effect, let's say, but sometimes an effect is not what you want. You see here the parameters that we measured, the respiration, fresh weight, shoots and roots length were not statistically affected except for cellular respiration. But let's say that as a starting point, the idea that the bacteria and the biochar do not impair the growth of plants is already a good starting point, because at least you know that you are not doing any damage. You never know if the biochar together with the plants, which is germinating, can exert, for instance, some germination effect. So it is always a good thing that you don't have negative effect or extremely positive effect. Maybe you don't expect them. So this is the basic starting point, and maybe I'm going too fast. However, the next step will be to determine what happens when we add the metals and metalloids, because we are focusing on cadmium and arsenic. Arsenic has been studied already in our laboratory together with different cultivars of tomatoes, so this is also a topic that we know about. And in our area, we have a problem sometimes of arsenic in tomatoes, because the soil is normally rich of arsenic, so it connects somehow also with ideas that can be of interest in our territory. So the purpose the next experiments that we are already doing will be to assess the effect of metals and metalloids alone, or in the presence of different doses of biochar. So I think this is all I had to do, all I had to show. The work in progress is, as I was telling you, that we want to develop these new strategies to understand what is happening in the plants. And we have to imagine also the microbes themselves. So there will be, for sure, some transcriptomics studies in the plants in different parts, roots, leaves, and pollen. Pollen, why? Because we recently studied also the effect of biochar on the reproductive capability of the plants. Metabolomics on the fruits, we have already done it in the field, so we want to check what happens to the food quality and the main metabolites of the fruits. Metagenomics, in order to understand the behavior of the microbes, but also of the rhizosphere, so not only bacteria but also fungal organisms. Of course, we will have to measure the metal and metalloid concentration, and the study of genomics in mitochondrial and plastid DNA, it also connects to previous studies that Luca performed with Jason and others, to understand what happens at the cellular level in plants treated with different kind of amendment. So I think I can go to the conclusion. Let's say we are trying to prove, because we have experience in the past, but now we have to understand what happens in this specific situation, to use plant growth promoting microbes as functional solutions for sustainable agronomic practices. We want to understand how the biochar functionalized with these microorganisms can become a promising strategy also for soil remediation if we can see that the effects by cadmium and arsenic are diminished. We hope they will not be instead increased. And we hope to find potential biomarkers for exposure and effect by studying the plant tissues and the fruits, which are, of course, in tomato very important, in condition of presence of metal and metalloids, and we hope positive effect of the treatment with biochar and plant growth promoting organisms. So these are the acknowledgements. The financing institution, which is the ministry, which, by the way, has not given yet €1, but of course they will, hopefully. And Consorzio Italbiotech, Consorzio Cinsa, and then the University of Parma and the Guangdong University. Thank you for your attention.
Thank you, Professor Elena, for very distinct speak. Now it is open for questions. Yes, please. Yeah.
Hi, Elena. Thank you very much. That was really interesting. You spoke briefly about the granularity or the granulometry of the biochar. Could you just give us a little bit more? What's the size of the biochar that you're using?
Okay, yes. So the biochar, as I told you, depends on the material, starting material, because basically the pyrolysis, or let me say, the pyrolysis in our special pyrolyzer machine that I did not mention, we use a prototype pyrolyzer machine working together with the company that holds the patent. In that case, what you put is what you get. So if you use pellet or chipped wood, you basically retain the same structure. Here I can see that there are no very big chunks, but they are maybe more small pieces of material. The impact is that sometimes in some pyrolyzer machine, especially the industrial ones, you get a powder. And with the powder of biochar, you increase the surface available, but you maybe lose some of the beneficial properties of the surface. No? So powdery biochar is also difficult to distribute because you breathe it, so you have to take more precautions, and you can use it as a slurry more or less. If you have chunks too big, of course, they will not be helpful because they will not mix well with your substrate, but when you have chunks, you can still grind them, so you can reduce the granulometry. Let's say that, especially if you use biochar as an amendment, also for the soil texture, this becomes very important. If you have a very compact soil, the chunks can help it breathe. If you have it very powdery, it gives texture to a soil which has not. So it is one of the critical factors that you have to choose. In this case, we used pots in the greenhouse, so everything more or less was okay. But in the field, it is important.
Yes. Thank you so much, Elena ma'am, for elaborating so much about the biochar. I just want to have one question that what is the mode of, is it used for in situ by remediation process wherever the contaminated site is there? How can you apply the biochar throughout the land? And what is the mode of optimization? For there are so many metals and metalloids which are being contaminated in the soil. Yeah. So how to optimize that? How much amount of, since it is synthesized in the laboratory, what I can see for pyrolysis process. So biochar is amended to the soil. So what should be the ratio of the contaminants to the biochar?
This is a good question because the application of biochar to contaminated soil is not so, let's say, not so well studied as the agronomic practice. In agronomic practice, you have some instances where you give the biochar before sowing or before transplanting, and in that case, you can mix up biochar very well, let's say in the first 25 centimeters, so they are in the root zone, and you can mix it with a machine or whatever. By the way, if you want to have the biochar as a carbon sequestration, you have to put it deeply because so it will not be disturbed. So if you use it for agronomic practice, it must be in the first 20-25 centimeters. But biochar is also applied in agroforestry where you have trees. So in that case, you cannot dig, you just give it on the surface and you make the appropriate calculations, which are not so easy to do. In order to mix it up loosely in the first strata. We have also applied, for instance, biochar in forests. And in forests, you mix it up with the litter and everything, so it can be applied in different conditions. Yeah, in situ, absolutely, yeah. The problem is, like you were saying, in comparison with the effect on metal, this is all to be studied. Let's say that if you use biochar as an amendment to improve the soil properties, you go at a high level. Somebody even say 40 tons per hectare, which is an enormous quantity. If you use it as a vehicle of microorganisms, you cannot functionalize 40 tons. So you go for much lower quantities. Let's say a catalytic biochar. So it just provides microbes and not so much an amendment to soil. So yeah, it is all still to be studied. And case by case, of course, every biochar, as very lot of biochar, have their own features and you must check it very carefully, for sure, yes. There is one there, but last one.
So just one small, as a query rather. So beautiful presentation, Elna. So what was the basis of this background for choosing 15 milligram per kg arsenic or 20 milligram per kg arsenic?
Okay, this is last question,
Yes.
and please reply
And
briefly.
the question is easy because Luca
And
Pagano
other
did it
colleagues can keep
and I
talking
don't
later.
know
So
why. I will ask and let you know.
yeah, thank you. Please. Okay, so now thank you. Let us give a big hand to Professor Elena for very vibrant presentation. Now next speaker is Professor Kundan Kumar. Professor Kundan Kumar is from BITS Pilani, Goa campus, and he is going to speak on an aquaporine member OST IP12 confer arsenide tolerance and reduce root to shoot translocation of arsenic in rice. So welcome Professor Kundan Kumar, and now it is yours time.
Thank you for introduction, sir, and good afternoon, everyone. And I would like to thank the organizing committee to giving me the time to present my research work. So I'll be talking about aquaporins. So aquaporins, they are involved in various transporting various molecules, including water, including urea, and various metals. So one of the member is OSTIP12, which I will be covering in this particular thing for making the arsenic tolerant rice as well as reducing arsenic content in the rice grain. So as we know that this is an old slide which suggests that arsenic contamination is there everywhere, including most of the contaminated part is South Asia as well as South America. And we know that this particular area is having a lot of arsenic contamination, mainly in the Bangladesh, West Bengal, and north part of India. So arsenic poisoning, you know that it is causing several types of disorders, including various types of cancers, skin cancer, and other types of cancer. And how it comes because of drinking of water contaminated with arsenic. Apart from that, it is also coming because of we are taking rice, which is somewhere it's contaminated, because you know that rice is that particular silicon transporter system is well established, and through that the arsenic enters into the rice, and it is the grains which we are eating. The grains which we are eating, that is contaminated with rice. That's why it enters into rice, and we are eating rice. That's why it comes into body, and that causes various types of disorders. So arsenic is stressing ourselves. It stresses plants also. So at various stages of rice, it is stressing the arsenic is stressing the rice, including germination. So you will find that germination is slower, or the reduced germination will happen because of higher arsenic content. Then vegetative growth will be slower, and decreased plant height, root width, and all those things will happen. Apart from that, reproductive growth will also be hampered because of arsenic stress. So coming to aquaporins are water transporters, and they facilitate transport of small molecules. This is a general slide which shows that how the aquaporins are transporting water as well as various other solutes. So apart from that, since the aquaporins are transporting, as we know that they are transporting silica, they are also transporting these metalloids, including arsenite, the trivalent form of arsenic, and then organic form of arsenic. So these aquaporins are well known. Some of the aquaporins are well characterized. So further on, these are the structure of aquaporins. So they have 6 helical structure, 6 transmembrane helix, as well as 5 loops are there, and there are some selectivity filters, including aromatic and arginine, amino acids, and Frogger position. And you can see that this is aquaporin assembly through which the arsenic or various other solutes can also get transferred. So coming to aquaporin classification, so you can see that they're classified into 5 different families: plasma membrane intrinsic proteins, PIPs. So my target was TIPs, because already there are reports which suggest that NIPs are involved in arsenic transport. So, and in my postdoc work, we worked on PIPs in Ohm's lab, where we showed that some of the PIPs are involved in arsenic transport. So I was wondering whether other members of aquaporins like tonoplast intrinsic protein, which is TIPs, which are present in tonoplast, mainly vacuolar membrane. So can they transport also arsenic? So putting this question, this can be answered. Basically, you can see that how we can reduce the arsenic transport in rice. So as we know that the various transporter like LSI1, LSI2, LSI6, they are known that they are transporting arsenic from the roots, and they are entering into the vascular system, and then through the vascular system it enters into the stem, through the shoots, and then it enters into the grains via various transporter like PTR7, INT, and PSTs. So this is how arsenic enter. Mainly arsenic enter through, in the grains it has been shown that INT4 and INT6 is involved in arsenite transport directly. So whether we can engineer those arsenite transporters, like we can knock out some of the transporter, or we can overexpress some of the transporter, which will make the arsenic more available to the roots, and they enters into the vacuole. So that is what we thought about, and that's how we thought of engineering those arsenite transporter in rice to make lesser arsenic accumulation in the rice grains. So this is a slide which shows that how aquaporins can be called as a metalloid transporter. So there are various reports which suggest that not only arsenic but also other metalloids like boron, silicon, antimony, apart from arsenic, they can also be transported via various transporter, including NIPs, PIPs, TIPs. So there was no report for TIPs at least. So we were lucky that there was no report for, but there was, during my research time, we found that one report came in Teres vitata, that is fern, where TIP14 was having, it was shown that it is causing the translocation of arsenic in the fern. So with that, this particular work was done in my postdoc work, where we have shown that OSPIP24, 26, 27, and PIP13 is involved in bidirectional arsenite transport. And with that, we have shown that in yeast system, as well as in oocyte system, as well as in plants, that these particular PIPs are involved in arsenite transport. Then further move on to TIP sequences. We cloned TIP sequences from rice, IS64, and we identify some of the amino acid changes were there. And we reported those changes which was not there in the plant. So much of the sequence was similar if you compare different subspecies of rice, but some of the differences was there, but those sequences not basically those which determine the selectivity filter, or those are which are required for the selectivity of arsenite. So we tried, so there were like out of 10 TIPs, we cloned 5 TIPs, and we tried to see that whether out of 5 TIPs, which TIPs will be responsible for arsenite transport. So we did, we had some mutant strains like W3031A, which is having functional FPS1, which is wild type. And YSH294, it was lacking the FPS1. So this FPS1 was lacking, which is a plasma membrane transporter. And IRW100, they lack YCF1 transporter. And IRW101, it is basically lacking both the FPS1 and YCF1. So basically, if you look into W303, which is wild type, it is having both intact arsenite effluxer and effluxer both, as well as IRW100, where the arsenite, the influxer of arsenite into, or influxer of arsenite into the vacuole which is involved in vacuole sequestration, that is absent. So we'll target only these two mutant strains, basically IRW100 as well as wild type strains. So we did East complementation studies. Since in IRW100 YSF1 was mutated, so can it replace the OSTIP12, which was the candidate gene? So can it replace the YCF mutations? So basically we found that if at higher concentration of about arsenite stress, you can see that when IW100 was co-transformed with OSTIP12, it was able to tolerate more arsenite comparison to wild types. So which indicate that these are transporting arsenite, and since these are transporting arsenite and it is entering into vacuole, that's why it is giving more arsenite stress phenotype. Basically, we can be able to get the growth of yeast cells at a higher arsenic concentration. And this thing was also done with the help of the OD, or the growth of the yeast strain, which was grown at higher arsenic concentration. And then further on, we looked at the arsenic content in both the strains, like wild type strains, and then IRW100 with empty vector, which is the lower one, and then the IRW100 with the OSTIP12 transformed. So the middle one will transform, so that basically this is complementing the effect of the YCF1. So we see that the concentration of arsenic was higher in the yeast strain, which was transformed with OSTIP12. So we thought that why it is higher, and since it is giving tolerance phenotype, but it is showing higher because the arsenic content is going into the vacuole, and what we are looking at here is basically entire arsenic content. That's why it is the concentration of arsenic is higher in the strain which was complemented with OSTIP12. Then we looked at other OSTIP members, which showed not much difference between the wild type and the empty vector as well as the OSTIP. So basically, out of those TIPs like OSTIP11, OSTIP12, 13, as well as other TIP member, we found that OSTIP12 is a member which is a target which we can target further, and we can go into the plant system. And we also checked for arsenate and boron stress, and we didn't found any much difference in arsenite stress and boron. That means it is having selectivity for arsenite only, not for arsenate as well as boron. So the cell survival assay, you can see that propydium iodine staining, it shows that at higher concentration of arsenic, that is 100 micromolar of arsenic, more deaths was there. That is what it will show. The propydium iodide staining was shown when it was inoculated with the empty vector. And when we transformed with the OSTIP12 with the IRW100, so you can see the lesser amount of propydium iodide staining was there, indicating the same thing, that when we complemented OSTIP12 with that particular strain, then plant were able to tolerate higher arsenite stress. And this was shown in flow cytometry analysis also, that about 91.3% of cells were live, and comparison to the other empty vector system, where 31.3% of the cells were live. So in aquaporins are getting inhibited via silver ions as well as sodium azide. So we have checked that whether this particular sodium azide as well as silver nitrate is able to suppress the effect of aquaporins, whatever the OSTIP is showing. So if we found that so this is arsenite content. So you can see that this is able to grow. But here at higher arsenic content as well as AgNO3 and sodium azide, the yeast cells were not able to grow. So it reverses the phenotype, whatever it was showing earlier. So indicating that whatever the OSTIP12 we cloned, that is aquaporin, and it is getting inhibited by sodium azide and silver ions. So a subcellular localization was done with the protoplast isolation, and then so the particular OSTIP12 was tagged with the GFP, and M-cherry is a vacuolar marker. So this OSTIP12 showed that it is merging with the merging effect it shows, and it shows that OSTIP12 is localized in the tonoplast. So it's a tonoplast intrinsic membrane protein, that is TIPs. So further on, we overexpress this OSTIP12 in Nicotiana tabacum, that is tobacco, and we did some studies with the overexpression of those OSTIP12. We have shown that the overexpression lines was showing better germination compared to empty vector as well as wild type, indicating that the germination rate has increased because of OSTIP12, which was transformed in the tobacco. And also you can see that wild type, this is overexpression lines and then empty vector. So plants at higher concentration of arsenic, it was showing better biomass, better shoot length as well as weight, as well as the root length also was higher in the overexpression lines compared to empty vector as well as wild type. So we did some analysis of arsenic content in the tobacco plant which was transformed, and we found that shoot arsenic content was lower, while the root arsenic content is higher in case of overexpressed lines. And similarly, the translocation factor was lower in the overexpressed lines, and it was higher in wild type and the empty vector. And similarly, BCF is bioconcentration factor. So basically, it was higher in the overexpressed lines, and it was lower in the wild type and the empty vector, which indicates that basically the thing is like the OSTIP12 is translocating the arsenic into the vacuole. That's why more arsenic is entering into the roots, and that's why it's not going into the vascular system, because lesser arsenic is there in the cytoplasm. That's how we can say that in shoots the arsenic content was lower, and in roots arsenic content was higher, and that was shown by a translocation factor. So similarly, this was tobacco. Then we thought that we should go in the rice, because the main target was rice only. So we overexpressed the same thing with the rice in PB1, Pusa Basmati 1, and we developed several transgenic lines of overexpressed lines. And here we did some rice seed priming with different yeast strains on arsenite tolerance pattern. So basically, the seeds were primed with those yeast strains. Those yeast strains was either empty vector, those yeast strains was either wild types, or the yeast cells with the OSTIP12 transform cells. So we found that in control there was no difference at all. When so basically after priming we kept in control conditions, and then we given some arsenite stress. So you can see that the PB1, which is without any yeast priming, so the PB1 was not growing well, and when they were grown with the OSTIP12, they were growing very well. So shoot length and shoot fresh weight was higher. So I will leave this. Basically, diathajon assay will suggest that how much amount of arsenic is going into the shoot. So we found that in overexpressed lines, more arsenic was there in the shoots compared to wild types. So you can see that in wild types, more concentration of arsenic was there in the shoot, in the roots, and basically in the shoot. And in case of overexpressed lines, lesser arsenic content is there in the shoot, indicating that more arsenic is there in the roots. That's why it's not going into the shoot system. So we also calculated shoot arsenic, root arsenic, and then calculated translocation factor, and we found same results how we got in the tobacco. So indicating the same thing, that it is happening the same way, like it is not, the arsenic is entering into the roots and it is sequestering in the vacuole, and not much arsenic is going to the shoots. That's why we are getting lesser arsenic in the overspace line compared to wild type, and more arsenic in the root of overspace lines. And translocation factor is also lower, like about 0.8%. And then we did some pot experiment to show that is there any difference between the wild type and overspace lines in the general or in the salts in the arsenite stress, and we found that in general in control conditions there is not much difference between wild type and the 3 overspace lines, but in case of arsenite stress conditions, the various types of factors like number of leaves, number of tillers, number of panicles was higher in overspace line compared to wild types. And similarly, we did arsenic in pot experiment also. The same studies was done, and in pot experiment also we used 20 mg per kg in pot experiment, and which show the same type of answer, like shoot arsenic is higher, lower in case of overspace line, and root arsenic is higher in the overspace lines, and in grain arsenic we have found that it was about both the lines was showing lesser, lower than detection level. So the hypothetical model which we explained from here, that the arsenic is entering via various transporter, and this transporter basically which is present in vacuolar membrane, so they are transporting with the help of that answer is not known, that how they are getting transported by phytokeratins or glutathione or metallothionein. And since most of the arsenic is translocated into vacuole, that's why lesser arsenic is available in the shoots, and that's how we are getting lesser arsenic in the grains. So this is the hypothetical model, and conclusion is like the OSTIP12 can be used as a system, and the overexpression of these particular plants has shown that it can take lesser arsenic in the shoots as well as in grains, and root to shoot translocation was reduced quite a lot in case of overexpression lines when OSTIP12 was overexpressed. And this is the acknowledgement to various lab members. Mainly the Suhas was involved in this particular work, and the collaborators were Dr. Sudhakar Srivastav and Suprasanna from BARC. And this particular project was funded by BRNS, and various funding agency has helped us, our lab, to grow. And so this is also, I want to we are organizing one conference, so that's this platform I want to show that we are organizing one conference in February. So we invite you all for presenting your work or oral presentation or poster presentation. So it will be 46th annual meeting of Plant Tissue Culture Association India and Symposium on Current Trends and Challenges in Plant Biotechnology, which will be happening in Goa. So Goa is a very beautiful place. It's almost similar to this particular place, but it's more vibrant, and you can come and enjoy the particular places. Thank you.
Thank you. Now the floor is open to discussion.
Yes, sir.
Beautiful presentation, Dr. Kundar Kumar.
Thank you, sir.
I think associated with Dr. Sudhakar Srivastav.
Yeah.
We work in the same group. So whether this transporter, which is an analog of the aquaponin, is similar to what is reported in the ACR3 protein in the form.
So
it is a variant, structural variant of that, because that ACR3 is absent in the flowering plants.
Yeah. So
it is localized in the funds only, in the hyperaccumulator, teres vittata, and those genes are absent in the flowering plants, for example in rice and many others.
Yeah.
So I want
so we have not checked ACR, but we have checked the PBTIP34, which is basically tonoplast synthesizing protein, which is only reported till now. So we have included that, and we have found that it is almost similar patterns are there, but the sequences are very different. But the patterns of the NPA motifs, and they are very much intact. They are similar to very much like NPA motif, as well as Frogger's positions are almost same. But we have not checked with ACR. ACR3 we have not checked.
ACR is absent in the flowering plant, that is established.
Yeah, so
it is just localized in the arsenic hyperaccumulator.
We have to keep the questions brief. Sorry.
I'm not sure whether.
Okay, thank you.
It's not a debate. Sorry for any other question.
Yeah, I think we are running with time, so
Yeah.
we can discuss it later if they.
Yes, we can discuss in coffee break. Okay.
The speaker is here.
There is no more questions. Okay.
Regions. The next was the task was to identify the domain. So NCBI conserved domain database and smart server were used for the distribution analysis of HMA conserved protein domains. The resultant files were given as input files to TB tools to obtain the domain diagram. Subsequently, the subcellular localization of HMA proteins were predicted using the Wolf PSOT protein localization predictor tool, and the resultant cellular signal values were plotted using Excel. Then the next task was to identify do the conserved motif analysis, which was performed by Meme, which is an online software for the production of conserved motifs. The distribution types of motifs were set to default parameters at 0 or 1 occurrence per sequence, and the number of motifs and the minimum and maximum widths were set as the values which are given in the slide. Then for the phylogenetic analysis, the full-length HMA protein sequences of Triticum aestivum, Oryza sativa, and Arabidopsis thaliana were used for the evaluation of evolutionary relationships. The multiple sequence alignment was performed using Clustal W and subsequently uploaded to Mega, and a phylogenetic tree was constructed according to the maximum likelihood method using 1,000 bootstrap replicates. Following that, the homologous and the paralogous HMA gene pairs were manually selected from the percentage identity matrix prepared using MAFT. The KS values were calculated using the TB tool, using the CDS sequences, protein sequences, and gene duplication pair as input files. For synteny analysis, it was conducted to find the evolutionary relationship between the wheat, rice, and Arabidopsis HMA proteins. So for that, the protein sequences of all the retrieved HMA proteins, they were used as input files to an online synteny viewer tool circulator using default parameters. So this first result shows the genomic distribution of heavy metal associated proteins in Triticum aestivum across sets of A, B, and D subgenomes. Next slide. Then the gene structure analysis result. So actually the tool was not able to take up all the entire 243 sequences. So I have just uploaded the sequences in a subgenome-wise manner. So this figure shows the introne and exone organization of HMA genes derived from the A subgenome of Triticum aestivum. And so that's how the genes are grouped. And we can see the legend which shows the CDS, the upstream and the downstream region, and then the introne and the introne phase in which the particular gene is possessing. And then this is a chart showing the number of genes present in the so this chart shows the number of genes present in the on the various on the monoploid number of chromosomes derived from the A subgenome. Next slide. So this is for the THMA genes derived from the B subgenome of Triticum aestivum, and subsequently from the D subgenome. And then we can see the number of genes present in the monoploid count of 7 chromosomes derived from the D subgenome, which shows the number of genes. Next slide. So the cis regulatory element distribution in 243 members of THMA gene promoters. So I apologize, there was no other way out. I mean, I'm still looking for a better representation method, by which we can publish the results in the manuscript. But this was something I did how I was getting the results from TB tools. But yeah, I've grouped the results as per the category of the cis regulatory elements that was found, that growth and development and light responsive, hormone responsive, and stress responsive. Next slide. So this is a pie chart showing the growth and development responsive, showing the various proportion of the cis regulatory elements found for growth and development, then hormone responsive, light responsive cis-acting elements, and stress responsive cis-acting elements or the promoter elements. So this is a Venn diagram showing set of the common genes containing various cis-regulatory elements. So about 193 genes among the 243 members identified in Triticum aestivum. This Venn diagram shows the 193 genes which contain the various cis-regulatory elements. Next slide. So this is a slide showing domain pattern visualization using the NCBI CDD tool. Again, a very space constraint diagram which is not clearly visible, but this is how I was able to fit in this slide. And again, a better method there should be some better way out there by which I can present these results. But I was on the previous slide only. Yeah, previous one again. Previous one. Yeah. So this pie chart, it shows the, I don't know why is it showing the, oh, yeah, that was something happening to me earlier. That's fine. So this is a radar plot for the predicted cellular localization, and the list of abbreviation shows the corresponding name for each of the organelles. So we can see a variable plot was variable cellular signal values were obtained for each of the HMA proteins, and they were plotted using this radar plot. Next slide. So this slide shows the analysis of motif diversity in HMAs. So again, the genes are grouped according to the subgenome from they are coming. So the first figure shows the motif distribution across the THMA proteins from the A and B subgenome respectively. Next slide. So this is for the D subgenome, and then the figures from D to K, they shows the sequence logos of the various conserved motifs found from the meme server. Next slide. So this is a phylogenetic tree which was obtained using the full-length protein sequences from using the 46, 55, and 243 full-length protein sequences from Oryza sativa, Arabidopsis thaliana, and Triticum aestivum respectively. So this tree needs still a lot of work, because there are, so these heavy metal ATPases, they are metal specific. They're heavy metal specific. So this tree would need work on grouping the genes based on the specific heavy metal they are involved in transporting. So this so I've just done the build the phylogenetic tree, but definitely this tree needs a lot of revision and lot of work in so that it is better interpreted for the evolutionary for understanding the evolutionary relationships. Next slide. So this slide shows the multiple sequence alignment, which was done using again a set of sequences of HMA from Triticum aestivum, Arabidopsis, and rice, and this asteristic marks show the conserved residues found along the sequences, and the highlighted regions. I'm not sure if they are very clear because I've highlighted them using a like faint yellow box. So they show the conserved residues and regions among the various HMA proteins. Next slide. So this slide shows the analysis of the duplication events. So this is the segmental and tandem duplication events detected for the identified THMA gene pairs. So various gene duplication pairs, they are connected by different colored lines, and they are connected on 14 wheat chromosomes. Next slide. So this box plot shows the KS and KS values obtained using TB tools. So this ratio values, non-synonymous substitution, non-synonymous to synonymous substitution ratio values, they are calculated using the full length sequences, and a value of 1 more than you can say more than 1 and less than 1, they indicate neutral, positive, and purifying selection respectively. Next slide. So this slide shows the visualization of the sequence similarity across orthologous gene pairs. So, so again, I have grouped it as based on the genes derived from a particular subgenome. So figure on the right shows the evolutionary link of rice HMP genes with the genes from the A subgenome of Triticum aestivum, and the figure on the left, it shows the evolutionary link of the Arabidopsis HMP genes with the Triticum aestivum, with the HMA genes from the A subgenome of Triticum aestivum. So the same colored lines, they indicate the syntenic relationships between the corresponding orthologous gene pairs. So, conclusion, so study at hand aims to provide a comprehensive in silico identification and subsequent characterization of HMA genes and its corresponding proteins in Triticum aestivum. So results so far have revealed a total of 243 HMA proteins. Then various other investigations they add considerably to the theoretical pool of knowledge for this crucial gene family in an important food crop like Triticum aestivum. So this is a future work that definitely would involve a lot of wet lab work and expression profiling and then functional characterization of selected genes. I would like to thank Guru Nanak Dev University, Punjab University, and University of Calicut for giving me maybe just a chance to present my work in a form of oral presentation. So thank you so much, everyone. And I apologize.
Yeah, we are a little bit out of time, but of course there were technical problems. So
I'm sorry
now
for that.
one or two questions. There? No, but okay. Women first.
Yeah, thank you, Mac. That's a nice overview of the entire family of HMAs in wheat. That's a good start.
Yeah.
But this is all in silico data.
Yeah.
So have you checked which genes, which HMAs are responsible for which heavy metals? Because you said heavy metals. That's a group of that.
Yeah.
And they might have different substrate specificity.
Yeah.
So then have you figured out?
That's what I was telling when I was presenting the slide on phylogenetic analysis, that this needs to be elucidated while in a phylogenetic tree, that which using the available literature for the that has been documented for the Arabidopsis model plants including Arabidopsis and Rhiza sativa, that based on that we can find those orthologous genes in Triticum aestivum, and based on that we can assign the substrate specificity, but that's just the bioinformatic work again, and it would involve the wet lab work for further validation.
Thank you.
Specificity.
There is another question, Jyoti, yes.
Thank you, madam. Nice presentation. I just want to know that out of 243 genes, what you have shown in your Venn diagram, that 193 genes that show some common relationships.
Common cis-regulatory elements, yes.
Yeah. So have you characterized, have you checked, in fact, the data in the gene bank itself, on what basis it is showing the common characters?
No, ma'am, I have not done that. So it was just that when I was doing the analysis for the promoter elements, that I was actually falling short of how to present this work in a better way. So this is how this question just came into my mind, that this could be an interesting way to put forward this cis element, this data on cis regulatory elements in the form of a Venn diagram, where I can see that which genes contain this majority of the cis regulatory elements. But yeah, this is something which just came in. This idea just came in my mind when I was doing the analysis, but I have not cross-checked it on any database. Okay. So yeah, thank you. Thank you.
I think we have finished with the question. You have your microphone open. Okay. So we can move to the next one. Thank again.
Now there will be presentation by two students, and they have to. These are for prize. These are prize presentations. There will be 15 minutes time per each. 10 minutes. After 10 minutes, there will be one bell, and then after 2 minutes again, there will be final bell. 3 minutes will be discussion. So please keep in time, because your evaluation will be done only when you will be able to complete your entire message within the time. So it is Sainika Jain is first presenter. She is from Banasthali Vidyapith, Rajasthan. The topic is arsenic-induced phytotoxicity in Triticum aestivum and probabilistic risk of carcinogenesis associated with consumption of contaminated wheat. So very important topic, and I hope the audience will enjoy it. Please.
It has been suggested that I give the chance for another question to the previous speaker. In my opinion, she already decompressed, so she doesn't want another question.
By mistake, she quit everything, even an online link. So the technical person has to come.
Okay, so what can we do? Okay. So even changing the speakers is not helpful because
we will keep the 15 minutes time. We will extend everything.
Okay.
Including dinner.
And what about the other session?
Others has the same thing. 15 minutes behind. So I'm coordinating both sides.
Yeah, you're right. Even delays must be coordinated. That's good.
They started late.
Oh.
But
Okay.
they are enforcing the time very strictly.
So we will keep the 25 minutes break with photo.
We will reduce that.
Reduce, not too much. Not too much. Just the photo will take 15 minutes. Then come back.
Grab the coffee, bring the coffee here.
Yeah, that's good. If we can.
And then somehow the AC is not working. Somebody's looking at that now. Only fans are working. It's getting hot here.
What can you do?
We are trying to get,
Oh, yeah, we have slides.
Tomorrow you will have coffee.
Okay.
We will ask them to have coffee. No, we will ask them coffee and tea.
So the good news is tomorrow there will be coffee.
I don't know.
In the break.
There's supposed to be a coffee as well, but I
Good. Are we almost ready? Everything is online, seems yes. Yes, okay, so again, thank you, and the floor is up to you.
Hello, namaste and vanakkam everyone. My name is Sanika Jain. I'm a research scholar from Banasthali Vidyapeeth. I'm doing my research on arsenic toxicity on Triticum aestivum, which is known as wheat. The topic of my today's presentation is arsenic-induced phytotoxicity in Triticum aestivum and probabilistic risk of carcinogenesis associated with consumption of contaminated wheat. Firstly, I would like to give a brief introduction about the problem, which is the heavy metal toxicity. Heavy metals are those toxic compounds which is being toxic at a very low concentration and when present in environment leads to toxicity to plants, animals as well as to humans. When present in agricultural land, it will lead to toxicity in plants and it left the farmers with poor quality of yield and severe crop damage. there are various sources of heavy metal in environment, which include vehicle pollution, air pollution, effluent release from industries. Amongst them, groundwater arsenic contamination is the major. So these heavy metals enter the food chain via plants because there are various vegetable as well as agricultural crops that has the potential to accumulate these toxic elements in their edible parts. So once enters into the food chain, it will lead to severe toxicity to humans as well as to animals. The acute diseases include headache, nausea, vomiting, and the chronic disease will lead to cancer and some neuro disorder. This is a world map showing the arsenic presence, which includes southern East Asia, and there, which include the Nepal, Bengal, India, Pakistan region, and China, Poland are also there, which is contaminated with arsenic. These are some newspaper article clips, which being published in prestigious newspapers, highlighting the presence of arsenic in groundwater. When present in soil, it will lead to toxicity in plants, will lead to crop production and stunted growth and poor quality of yield. So I've taken Triticum aestivum as my experimental plant. It is a 1.5 meter tall annual plant and can withstand severe winds. The crop is very rich in protein, carbohydrate, and vitamin, and it is the second most important crop across the globe. So my presentation is divided into 3 parts. In the first part, I'll be talking about the toxicity related to arsenic on wheat. In the second part, I'll be talking about the probabilistic risk of carcinogenesis. And the third part, I'll talk about the mitigation approach. This was the methodology I have used. The methodology begins with the pot experimentation. I have carried out in vitro pot experimentation, and for my research work, I have taken 3 varieties of Triticum aestivum, which were HD2733, HD3177, and HD2824, which is majorly cultivated in arsenic-contaminated regions of India, which is Assam, West Bengal, Uttarakhand, Jharkhand, Odisha, etc. Then after 30 days and 60 days, arsenic accumulation was analyzed in root and shoot. Biochemical tests were performed. Its localization was analyzed by microscopic analysis. Then the accumulation in grain was also evaluated. Probabilistic risk assessment was evaluated. And lastly, I have come up with the solution using the nanoparticle mediated crop improvement. Here are the results. The soil that I have used was very fertile and rich in micro and macronutrients. As you can see, with the increasing concentration. So for my research work, I have used a range of sodium arsenate. I have spiked the soil with known concentration of sodium arsenate. The spiked concentration was 10, 20, 25, 50, 100 ppm. And you can see the germination graph. It was declining as we increase the concentration of sodium arsenate. The pot experimentation was carried out. The plants were given 13-hour photo period. So it is the morphological analysis. At 60 days, you can see in the picture, there was a reduction in shoot length and root length also. The accumulation was evaluated by using the atomic absorption spectrophotometer. This is a graph of AAS analysis in which the highest accumulation was found in root than in shoots. Proline and polyphenols are also act as some antioxidant, and it increases their concentration in the stress condition. 4 antioxidants enzymes were analyzed: catalase, ascorbate peroxidase, superoxide dismutase, and glutathione reductase. The graph showing the increasing percentage of these antioxidants with increasing concentration of sodium arsenate. So I've done some secondary metabolite analysis also, in which I've performed thin layer chromatography. The bands were observed for 4 secondary metabolites, which were gallic acid, salicylic acid, rutin, and quercetin. I've observed some dark bands and light bands in the case of stress treatment. To validate the secondary metabolite presence, I have analyzed the sample using the high performance liquid chromatography, and the peak showing here validate my results, and in the treated samples I have observed the reduction in the peak area and low concentration of secondary metabolites. I've observed the localization of arsenic in plant tissues. These are the images of light microscopy. This is the root section, and this is for the shoot section. The validation of the microscopic data was done by scanning electron microscopy. We have the FSM EDS spectra. In the EDS spectra, arsenic peak was present. This is the FSM EDS spectra of control shoot of treated and control plants. So after that, the agronomic traits were evaluated. These were the agronomic traits that I have observed, and I've observed that all these parameters were reduced with increasing concentration of arsenic. This is a picture showing reduction in spike length. You can see the picture of grain, comparative data of control and the treated sample. The treated sample, the grain of treated sample was appeared to be deformed with less nutrition. The treated sample, the treated grains were allowed for the regermination percentage, and it was also in the decline mode. The accumulation of arsenic was also, the accumulation of arsenic in grains was also analyzed, and I found that 0.8 mg per kg arsenic was detected in the grain. So it is the problematic condition for a crop plant to accumulate such toxic element in their edible parts, because ultimately it will enter into the food chain. So this is all about the toxicity. Further, I want to check the nutrient content in the plant. So proximate analysis were done, highlighting the testing of moisture, ash, fat, fiber, and protein and carbohydrate content. So comparatively, the nutritional value was degraded. 4 micronutrient content were also checked. That also validated my data that it was less nutritive. So this toxicity will lead to the condition of malnutrition. So to create awareness about this problem, I have done risk assessment, in which I did non-carcinogenic risk assessment, which is denoted by HQ. I have calculated the value for the different concentration. If the HQ value leads the value of 1, it will be subjected to carcinogenic. So after calculating the carcinogenic risk, I have concluded that all the treated samples accumulated the arsenic was found to be carcinogenic. So all these problems comes with the toxic condition of soil. And as Dr. Vara Prasad sir said that if we do not have the healthy soil, do not have healthy plant, and ultimately we do not have the healthy human and healthy environment. So it is better to remediate the soil first. So I agree with your point of view, sir, and I've come up with the solution of using nanotechnology, in which we synthesize some nanoparticles in nanoformulation, nanocomposite. I have synthesized iron nanoparticles by the green remediation approach. So this is the mechanism of iron nanoparticles. We incorporated the iron nanoparticles in the soil. When arsenic comes up in the soil, iron nanoparticles binds to the sodium arsenate. It has the potential to bind to both of its form, sodium arsenate and sodium arsenite, and form the Fe plaque. So this Fe plaque will not let the arsenic available to the plants. And the iron leads to the arsenic phytostabilization and leads to iron-rich and arsenic-free grains. That's how I have synthesized the nanoparticles. The size of the nanoparticles was 50 nanometer. I've given treatment to nanoparticles, and I've concluded that with the treatment of nanoparticles, the plant growth were enhanced in all the 3 varieties, as you can see in the picture.
With my research, I've also found that at 100 ppm concentration, I didn't found the presence of grains. But after the incorporation of iron nanoparticles, the presence was there. So by the incorporation of nanoparticles, all the agronomic traits and nutrition contents were enhanced after 120 days. These are the conclusion of my result. All the 3 varieties were found to be arsenic tolerant. Arsenic content in grains were found to be above permissible limit. And by using the green nanoparticles, it will enhance the growth and reduces the arsenic accumulation in root, shoot, and grain, hence contribute towards the sustainable development. These are my publications. These are my awards and achievements. I would like to thank Professor Ina Shastri, Vice Chancellor of Banasthali Vidyapeeth, Dr. Jyoti Mathur, my supervisor, Dr. Om Prakash Dhankar, and Dr. Joss Puthur, coordinator of IPC 18 conference, for giving me the platform for presenting my research work. Thank you so much. Thank you, audience, for patiently listening to my presentation.
A very nice presentation.
Thank you, sir.
So what is the pathway of arsenic uptake in the wheat?
Sir, as far as concerned, that arsenic is an analog of phosphate. So it competes out the phosphate in soil and taken up by the root cell walls, and by the xylem vessel, it's sequestrated and gets into the vacuoles, I guess.
Besides phosphate, is there any other route?
Just one question, please. Okay.
Okay, I have a follow-on question.
There is a question there, Liz.
Yes, yes. Thank you for your lovely talk. Iron has competition with other elements for transporters. I know there's a cross-talk with copper transporters. So did you see any differences in copper uptake when you treat with iron?
No, ma'am. No.
Jason?
Yes, sir.
Very nice presentation. I was actually going to ask something somewhat similar. Did you see any increased iron concentration in your grain?
Yes, sir. Yes, sir. Iron
Okay.
concentration was enhanced after the incorporation of nanoparticles, iron nanoparticles.
Okay.
Yes.
Because Elena likes me, she's going to let me ask a second question.
If it's cleverly done, yes.
I'm just curious, do you think you would have seen a similar effect if you just added iron sulfate or some conventional form of iron? Does it need to be an iron nanoparticle?
Sir, I didn't get your question. Can you please repeat?
Do you need to add iron in a nanoparticle form to get the reduced arsenic uptake? Could you just add iron
Yes,
sulfate?
sir. I've used the precursor, which was the FeCl3 salt. Iron salt was there. I used the iron salt for reduction of iron in the aqueous solution for the synthesis of nanoparticles, iron nanoparticles.
Okay, but if you had just added the precursor instead of the particle?
To move.
No, sir. The precursor was added to synthesize the nanoparticle. I've used the tea extract, which is having the iron in it.
Okay, we can chat later.
Okay, yes, we must stop. Thank you, Jason. Thank you, Sari.
Thank you.
And now the last but not the least, Jose Angel, Mexico. And he will discuss potential use of cadmium-tolerant plant organisms for artificial wetlands.
Hello, everyone. I'm glad to be here in this far and beautiful country. Yes. Well, you already heard that. Potential use of cadmium plants can absorb heavy metals. This is an important topic. I can just... Okay, so I need to ask to change your...
It's a PDF, so you cannot change. You have to move it.
That's the problem.
It's not a PowerPoint. Maybe. Okay. this
computer man.
good.
It's great. Yes. Well, let's talk about humans. Every needs every high amounts of different kinds of elements. So they continuously dig in the earth crust to find them, and only obtains little traces of these parts. But in the future, a lot of these elements could return to the earth, but in a potential toxin forms called PTE. So as a result, cadmium was chosen because it's a great indicator of contamination. Well, in the minery process, the minery industry used some acids to wash the rocks and then separate the element interest. But this creates acid drainage, and this acid drainage could go directly to the aquifers, rivers, and lakes. And then this water could be used in irrigating fields. So it's too important to understand the complex of this element. Cadmium has no use in the nature. Some bacteria, some another kind of plants don't use it to be a generator of biomass and produce energy. But the humanity uses this to create batteries, stabilization of metals, plastics, and paints. So, what's the importance to study cadmium? Because the cadmium is a dangerous element, can produce cancer, but also the most important illness, cadmium can replace the calcium in the matrix calcium in the bones. Basically because the calcium and cadmium has the same electron valency. It's like has the same size. So the calcium cannot join bones with the calcium, so it creates an illness too similar with osteoporosis. This problem was occurred in Jinsu River in Japan, and the illness is called itai-itai. In English, it's heart. So that's important. We create a lot of mechanisms to immobilize in the soils. Vitrification, pyrolysis, often with organic solvents, but then there are expensive methods. Take long time, many energy. So even the bacteria need special conditions, anaerobic conditions, pH levels and temperatures to 25 to 40 Celsius to be operated. Well, the bacteria uses the cadmium as final acceptor of electrons in the sulfate reduction process, but the bacteria needs a specific condition. So that's the reason why you need to use plants, because the plants can survive in any kinds of environmental, don't need any specific temperature conditions. Well, maybe we cannot put plants in the Antarctica or in the Pole North, but in some places the plants can survive to different soil conditions. So this is a small list of many of the most used or most studied plants. All of these plants are already studied. But we know that every plant in this planet can absorb heavy metals for this evapotranspiration process. But this starts a great way, a new researching line to find another candidates. We started to the most famous, for example, in China. In China has a lot of these problems, irrigation. The mining activity continuously giving to the Chinese population a lot of water. So in this researching, we found that 37 milligrams per cadmium per kilogram in the soil was there, but the plants, cabbage and spinach, absorbs similarly, for example, 12 to 15 milligrams per kilogram of soil. Too similar, the concentrations in the soil and in the plant. It's a problem because cabbage and spinach are the most consumed plants in the world. Even in Mexico, we consume a lot of spinach, so it's so dangerous. Well, for example, here, using atomic absorption, in Colombia, the researchers found, put these plants in 0.24 milligrams of cadmium in liquid media. But then they found that even in the plant, absorbs more than in the concentration. That means that the plant, before often directly of the nature, the plant already are contaminated with metals. For example, this is a great example of hyperaccumulator. Salsoca cali, this is a real plant. I never hear about it. But this plant can absorb, well, after this acid dissolves, the researchers found that in the plant, exactly in the root area, 2,696 milligrams per the cadmium are in the root system, is a lot of cadmium, and the plant could just continually surviving with nothing happens. Also in tomato, in Turkey, the researchers found concentrations of 0.12 milligrams cadmium, but no cadmium was found in the fruits. So the plant sacrifices itself only to the fruits can survive and not absorb heavy metals. It's a good topic. For example, in Africa, in the carrots, continuously absorb concentrations of 0.48 micrograms and 0.66. Well, the question is, all plants, all the plants we consume can absorb heavy metals. That's the importance to study this. Well, the reed in Mexico called carrizo is a cosmopolitan plant. Many research found that this plant can absorb lots of cadmium, 93% of efficiency absorbing cadmium in the media. In Italy, led these plants in a hydroponic system for 21 days. And after finish this experiment, the plant could continuously normal, maybe with less photosynthesize production, but the plant continuously goes very well. So in Mexico, we are working with the cells, with the most elementary part of the plant, called meristematic cells. These meristematic cells we can often, the entire plant, we just put another proteins and another enzymes to the plant can continuously growing. But the cells, we found this. In the cells, this is zero concentration. This is the chloroplast, are already function, but in concentrations up to 2.5 milligrams of cadmium, after tinted with blue methylene, we found these black areas into the cell. This is where the plants could grow in size, in get more bigger, because the cells expands. When the cells are exposed to a high concentration levels of cadmium, the cells just growing, and this black area, the next month when I return to Mexico, we take these samples. I'm go to analyze in a spectrophotomic absorption to determine how much cadmium is in there. So a lot of mechanisms are described in literature, but especially the plants produce a lot of enzymes in the root system when the amounts of cadmium elements are present. One of these enzymes, well, it's not an enzyme, it's a tripeptide, it's called glutathione. Glutathione is the most important because it's composed by glutamine, cysteine, glycine. And when these molecular assemble join with other enzymes, could produce glutathione peroxidase. This is responsible to bring the cadmium directly to the roots into the cells. Well, this is a process when the oxidative stress occurs into the plant, produce a lot of radicals. These radicals can be transformed to peroxidase. And then this peroxide could go directly with the ascorbat glutathione cycle into the cell and actually interchange with water. The oxide peroxidase could transform to water, and then the cadmium go inside to in the vacuole. This is not new. This has already happened in Bolgheli Lange in 1999, but always the images there was so old. And another example of this are the corn has these similar mechanisms. Even in the rice don't have this capacity to get the cadmium into the vacuole. It's only in the external in the cell. And this is an example how these cations interchange occur in the root system. It's too important to mention that it's necessary in the soils. The pH need to be acidic. If it's basic, the cadmium are immobilized. This needs to be acid, the soil, to the plants can easily obtain and extract the cadmium. So this is a little examples of how the cadmium could affect the plants. Decrease of the mass, decreases in the photosynthetical production, inhibiting celling, germination, stomatal and close of this evapotranspiration process. So to use all of these plants is necessary to put in acidic conditions, and the most efficient organisms are lettuce, spinach, carrots, reeds. Even the reed is an semi-aquatic plant, and in the aquatic media, the interchange will be faster than in the soil. So that's a lot of plant organisms could absorb these heavy metals from the soil, but this is actually when in the cells happen this process. The cadmium are compartmentalized, and then the cells dying produce new cells without cadmium. So in Mexico, we just trying to find if to make this new biological clean process from the soils, we need to ensure that the little ones, the older plants, can survive to conditions like 400 milligrams per cadmium. But we need to ensure if the youngest can survive in the same conditions. If the youngest plants cannot survive in these similar conditions, we cannot ensure if the reproduction process could continuously happen. So that's why the recommendation is to study not only the biggest plants, we need to study the embryons, the little ones, in the same conditions to be ensured if these plants could continuously reproduce in contaminated soils. So this is the recommendations, and this is a wetland in Mexico. In Mexico, we used to use these wetlands to clean a lot of water using this plant called reed. But many soils there are too contaminated by mining activities and produce a lot of this cadmium. Well, I will thank you to listen this. I think I will be faster than I expected, but I just want...
Time's up.
Okay.
The question?
Yes, question there.
Okay, so thank you, Jos, for presenting a nice research work. But I just want to know, out of all the slides what you have presented, which work you have carried out? Is it your work, or it is only the review of other papers which is published based on
sorry.
Yeah.
Yeah, it's a record of exactly is like how many plants can absorb heavy metals and the most important ones. A lot of researchers around the world study it continuously, but this work is reduced and only contains the most important for many applications. Persons who are not scientifics could learn about these plants and make these correct decisions to put this in contaminated soils. But also, it's too important to ensure where is the source of or water. You know, we cannot ensure if your water uses in irrigating fields are clean. So that's the purpose of this, to make this reduction of information. And
hello, her question is, what is your work? What is your research work? Out of your presentation, what research work has been done by you?
Oh, here. I'm working with reef cells plant, with a reef plant cells.
This study has not been elaborated, and the conclusion to this particular study, mostly it is all reviewed from the previous data. That's why I asked this question. Mostly slide is focused on the previously published papers, and reviewing that
of
data
course,
only,
yeah.
it was very less. Your own data is actually not concluding anything.
Well, the conclusion is to know how many plants can absorb heavy metals. And the most usually is carrot, lettuce, spinach. Maybe some people don't know that the plants could be dangerous if we don't know the source of this.
Thank you. There is another question. There, please.
Hi. So glutathione has a lot of sulfur in it. What's the effect of a sulfur amendment on the cadmium resistance? Have you checked?
Yes, the sulfur can be can bear the absorption of cadmium, because the glutathione in the ascorbate glutathione cycle use this peroxidal oxide, and the sulfur contained in the salts in the soil is like making a cofactor to increase the absorption of the cadmium with these peroxide oxides, and then the sulfate can go directly into the cell and making this interchanging into the cell. The sulfur is too important because sulfur are in every part of the world, but is a cofactor to increase the interchange into the cell to exit the cell.
What is the permissible level of cadmium?
Okay, madam, please.
Yes. Given by
No, I think we need to close. Okay, I understand.
Thank you.
Because
Thank
she's
you much.
evaluating, but okay. Thank you to all the speakers, and we really have to move to the photo.
First, there will be tea, and after quick tea, we will be going to the on the other building on the stairs for the photograph.
Okay.
On the balcony in the physical education building.
Gracias.
First. Which next sessions? Come up with the next session.
Next session, we have to take this pen drive.
Session 2.
Yeah, session 2, which one?
No, not session 2.
It's not here. We have to take this pen drive. Can you please check that? Yes, ma'am. Yes, sir.
This is one pen drive which is.
You share your information in the registration. Yes, so it will be. Yeah. Yes.
This pen drive is which one?
Okay, sir.
This is which pen drive?
This first one. You have to put it. Yeah. Yes, okay.
At many places, including National Botanical Research Institute. And I invite Professor Dhankar to give his presentation. Professor Dhankar. You want to speak something?
Thank you, Dr. Tripathi, for the introduction. This is basically, I put this talk as a filler because there were some empty slots, and as an organizer, I try to fill the slots with either my talks or some other my colleagues that I know that can do second talk. Anyway, so this one is old work with some renewed interest, and that's how I will be showing you with that. Okay, so in this one is the environmental mercury problems, that what are the problems of the mercury in the environment, and then also what are the solutions, and are there any the strategy that we can use those to clean this the mercury contaminated soil. So I will show you some of the work that has been done with using the bacterial genes, and then transferring those that strategy from the Arabidopsis to the real field crops for mercury and other heavy metal detoxification and phytoremediation. And then I will show you some of those, the nanosulfur, how the nanosulfur can alleviate the toxicity of mercury, and that will be for food safety purpose, not for the phytoremediation purpose. So global mercury pollution, mostly the major problem of the mercury is the artisanal mining. Around 38% of this is artisanal mining here, and then these other sources are basically the burning of the coals and other cement production and so on. But artisanal mining is the big one in the environment, because they use mercury to mine for gold, because mercury react with gold to make amalgam. And mercury problem is global, has a big problem. And also because of the many human activities, as well as the burning of the coals and mining and other areas. And mercury species, like organic form of mercury, methyl mercury, is highly neurotoxic. And then the ionic mercury and metallic mercury are also toxic. And also there are all these kind of the coal burning power plants, mercury in the bulbs, and the mercury in the seafood, and so on. And there is also a historic point of view, that is mercury, the Mad Hatter syndrome, that because they were using the mercury for shining the felts in the hats, and then the workers were exposed, and then causing the kind of their dizziness and moving in an uncontrolled manner. That was because of the central nervous system was damaged. And also there is a historical point of view. Mercury is Minamata Bay in Japan, that was in 1950, that was detected. That was the biggest disaster of the mercury contamination, consuming the mercury contaminated seafood from the bay. Because of the power plants and all those, they are dumping the mercury in the bay, and people were eating those. And those, like 2017, there was a Minamata this convention basically on mercury, and there is a memorial in there that these small dots, they represent the mercury, these balls. Some of you might have. When I was in high school, nobody knows whether mercury was toxic. We break the thermometer, and then we just spilled on the tables, and then we were having fun on those, but those were the mercury kind of these balls. And there are miners and all those. There are mercuries in the teeth feeling as well. And when your mouth is basically hot chemical factory that go cold or hot, and some of the mercury is basically released from the oxidation reduction reactions, and get into the guts and affect your microflora in the guts, and affect the health as well. And artisanal mining is basically biggest problem. There are more than several millions artisanal miners globally in South Africa and South American countries, Brazil and all those. And that's basically they're penning the mercury by hand, and then every 1 ton of gold, maybe around 2 tons of mercury was released in the environment as well. Mercury is extremely neurotoxic and causes the central damage to the central nervous system and many other health defects as well. And when the mercury is contaminated, and any other heavy metals, they are contaminated, either the water or the soil, and then these toxic metals come in the food chain and affect our health as well. So in the mercury, basically, mercury is the 3 form: methyl mercury, organic form of mercury. And organic form of mercury is converted basically to mercury 2. Ionic mercury. Ionic mercury is converted to mercury 0, which is metallic mercury. And there, in the plants, there is no mercury detoxification system so far yet detected. And and there is no mercury hyperaccumulator plant has been identified. So there is no solution that we can use plants to detox for phytoremediation of mercury or in this one. Bacteria has evolved these two genes, mercury detoxification pathway, MER-B gene and MER-A gene, and those convert this methyl mercury to mercury 2 and mercury 0. So with that one, this is I'm recycling some of the old work when I was in my previous life before my academic life in my at UMass. It was work from my postdoc days with Richard Meager lab, and so this is the pathway, and we put these two genes in the plants individually as well as stacked together, and then what happened there is basically when we overexpress the MER-B gene from the bacteria to the Arabidopsis, these are the wild type plants here, MER-B plants and then MER-A plants, no mercury. And when we grow on a 2 micromolar methyl mercury, the wild type plant did not germinate, MER-A plant did not germinate either because they cannot convert methyl mercury to mercury 2. But the MER-B plants that convert methyl mercury to mercury 2 and to mercury 0, that's basically they thrive on that one. And here when we overexpress the MER-A gene, the wild type plants were not able to germinate, seeds were not germinating. The transgenic lines at 50 micromolar mercury chloride, they are highly tolerant and resistant to that mercury. And when we put these two genes together, you can see this, this is the wild type, then MER-A, then MER-B, and then MER-A and B. And when we grow those without any mercury, when we grow those on the different concentration of mercury, as the concentration of mercury, methyl mercury increase, the MER-A and MER-B plants expressing together, they were highly resistant as compared to the wild type and MER-A, and even MER-B alone were not as tolerant. But MER-A and MER-B together, because that the entire pathway converted methyl mercury to mercury 2. Mercury 2 is converted to mercury 0, and mercury 0 is in the volatile form, and that released in the air. And that's how the plants are detoxifying that one. So based on that one, we basically developed a strategy that we can put those two genes together and plants can basically blow the mercury in the air. But that's not an ideal strategy either, because in the highly populated area, people will be breathing the mercury fumes. That's not an ideal solution. So we need to find ways to how can we convert the methyl mercury to mercury 2, and then how can we retain the mercury 2 in the plants for accumulation and harvesting and recycling and cleaning the environment. So that's where also we, because many of these underground aquifers were contaminated with those mercury as well, and these plants, the cottonwood, the trees can, the roots can go to 30, 40 feet deep and can pull out the water because they are the basically hydraulic pump. They can transpire almost 300 gallon water a day, or more than 1,000 liters a day. They can transpire that, so they can pull out more mercury and convert into mercury 0 and release into that. So in the unpopulated area, thinly populated area, that strategy can work. But mercury ultimately will come from the atmosphere back to the earth as well. So it's a cycle will be keep going. So then we transfer those the genes from the bacteria into cottonwood in the trees, and here is the wild type and MER-A plants without any mercury in the and that when the control condition after 14 days 300 ppm micromolar concentration of this the mercury, you can see after 14 days the wild type plants, they're dead. Transgenic plants are still growing healthy. They are converting mercury 2 to mercury 0 and growing in that one. So at the time, so it works very well. These plants can be effective, but we want to retain the mercury inside the plant so that we can harvest the plants and recycle the mercury from the plants and clean the environment. So in that way, we need to do the hyperaccumulation in the above-ground part. So what comes the handy is the glutathione detoxification pathway. I've been working on this pathway since my postdoc time, and this is we are still working on this. Tomorrow's talk will be focused on the glutathione homeostasis and how that will affect the heavy metals and the abiotic stresses. So in this one, the first gene is the gamma-glutamyl cysteine synthase that converts join the glutamate and cysteine to make dipeptide, the glutamate cysteine gamma-EC, and then the glutathione synthase add glycine to make tripeptide glutathione and phytokilatins. And here when we overexpress the first step, the gamma-ECS is the rate limiting step. When we overexpress that, the plants become highly tolerant. And here you can see wild type plants and gamma-ICS plants, and without mercury. With the mercury here, the wild type plants, they don't grow. Gamma-ICS lines expressing this gamma-ICS line gene lines are highly tolerant to mercury chloride. And but we want to basically trap mercury in the plants. So we combine the gamma-ICS gene and MERB gene, because MERB will convert methyl mercury to mercury 2. Mercury 2 is thiol reactive that can bind to sulfhydryl groups or sulfur groups in the glutathione. And once it bound, that glutathione mercury complex will be transported to the vacuoles for storage, and then plants, that's how the plants will detoxify and hyperaccumulate. So we overexpress those two genes, and then plants become highly, highly tolerant here. And then we did the Arabidopsis. Then in my lab now, currently what we are doing is we are basically transferring this technology to the non-food, high biomass, fast-growing field crops for phytoremediation of mercury. And we settled down on Cranberry absinica, which is a non-food, non-edible, and also short life cycle for maximum growth is for foliage growth, vegetative growth is around 45 days. And then we can grow multiple cycles in a year for phytomedicine purpose. Also, the good part of this cranberry is it is basically does not cross-pollinate with any of this mustard species. It's strictly self-pollinated, so there is no gene flow concern from that as well. So what we did was here, something wrong. There were some slides here missing. I don't know what happened. Anyway, I can just skip that. So what happened here in cranberry, there were some two slides that missing. We basically use this same approach. We put this gamma-ICS gene and MERB gene individually and combined, and this cranberry become highly, highly tolerant and accumulate very high level of mercury. And that paper is actually in Plant Cell Report this month is coming now. It's already online, I believe. Okay. So then what we did was, that was phytoremediation approach. But what about the food safety? Because mercury is coming into the environment and the food chain, and that's affect our health. So we were looking at a non-GM approach. Can we use the nanotechnology to rescue and restrict the mercury into the food chain? So we use nanosulfur, and we use we expose the mustard plant with a different form of nanosulfur, and bulk sulfur and ionic sulfate, and then we use those to see the growth. Even these sulfur forms are basically acting as fertilizer as well, because extra sulfur will enhance the plant growth. But when we grow the plants at different concentration, like here we use around 120 micromolar mercury chloride or ppm of the mercury, and we use this one with the different nanosulfur forms, and you can see with the 100, 200, and 300 milligram per kilogram of the sulfurs, and with the 100 milligram per kilogram or 100 ppm mercury chloride, you can see plants with a nanosulfur, they have the highest biomass. Here they were not inhibited, the growth was not inhibited, and you can see with the 100, 200, and also with the root biomass as well, as compared to the bulk sulfur and ionic sulfur. So nanosulfur is increasing the mercury tolerance in plants and enhancing the plant growth. Why? We look at the mercury in those plants, like here you can see this, here in this one, the mercury in the root content was significantly decreased with the nanosulfur. In the root content, the mercury in the roots was significantly decreased as compared to the ionic and bulk sulfur. Same thing in the stem as well, in the shoots also it was significantly decreased. So this shows that nanosulfur is effectively inhibiting the accumulation and uptake of mercury in mustard. And most likely now we are going to do the speciation. What we are doing is most likely is the mercury complex with sulfur in the soil, making mercury sulfide complexes, and that's basically mercury become insoluble and not bioavailable to plants. And then we also did using this different form of mercury, we look at this, what fraction of mercury, most of the, here in this one, you can see nanoscale basically decrease the mercury, the bioavailability of the mercury in the soil, because mercury become insoluble in that and less available to plants. We also look at we did the RNA transcriptome. That I'm not showing it because of the time, but also we look at the effect of the microbial diversity in the soil with the nanosulfur and mercury. And most of these beneficial microbiome were basically enhanced with the nanosulfur, and that affect those this plant growth and improve the plant growth and make the healthier plant. So with that, in a quick snapshot, I showed you that there is no solution for no phytoremediation or phytotechnology approach for plants. There is no plant to be identified as a mercury hyperaccumulator, or no mechanism is known in the plants. So bacteria has evolved with the mechanism of detoxify mercury. We borrowed the genes from bacteria, engineered into the Arabidopsis. We optimize those, what works and what does not work in Arabidopsis, and then we transfer into the high biomass cranberry for field application for phytoremediation of mercury. And then we are looking at for food safety purpose to inhibit the mercury accumulation in the plants, so that using the nanotechnology approach, and nanosulfur has been effectively shown that not only mercury on this talk, but also we have published a paper in rice as well. It 75% decrease in arsenic accumulation as well in that. And so that's basically the mercury is there is a strategy for mercury that we can enhance, and we can develop plants for that. So this work was basically done by my former postdoc in cranberry. This work was done by my former postdoc Sudesh Sikara, and now the current postdoc Yogita Singh, and she's basically working on those combined pathways, and Richa is putting several genes together for arsenic, and these current students Joe and some other. And some of those, Kundan also did some of the work in my lab a while back for the arsenic detoxification in cranberry. So with that, I and my collaborators, and several are sitting here in the audiences as well. And with that, I will stop here. Thank you very much.
So, thanks a lot, Professor Dhankar, for wonderful presentation.
I did it in 17 minutes.
Yeah. For that, I appreciate that for keeping the kind limits. So we can go for a couple of quick questions regarding his presentation. Yeah, please.
Quick one. Has anyone looked at sulfur-reducing bacteria in the rhizosphere? Do they contribute to reducing?
Yep, there is a direct impact of the sulfur-reducing bacteria in the soil. But for the plants, the bacteria is doing both ways. Bacteria are doing the demethylation of the mercury as well as methylation of the mercury. They are interconverting the methyl mercury to ionic mercury to metallic mercury, and back and forth. So it's basically they are doing both ways. So there is no net gain for the plants because mercury is floating in all three different forms there. Yeah. And at the ocean floor, the sulfur-reducing bacteria are playing a big role in that mercury geochemical cycle of mercury as well. Yeah.
Any other question?
Yeah.
Please.
Yeah, you use the mic there.
So because you said the microbiome has changed and there is an importance of the beneficial bacteria which is growing better, did you check what kind of bacteria are this? Any idea?
Yes, we did that. I did not show that. Here is all these different kind of the classes of the bacteria. Because of time, I did not highlight that one.
Okay.
Yeah, we did that one, all of those. Paper is already published actually in the International of Phytoremediation in 2024, at beginning of that. So I can forward that to that link to that one. I know your advisor, so I can forward that. Yeah.
Thank you. Thank you, sir.
Yeah.
So whenever we are working for this remediation approach or even for the transgenic approach for mercury compounds, so methyl mercury is going to be more toxic than general mercury which is being effluented from various industrial sources. So I think for such kind of approach, we should try for methyl mercury compound rather than the original one, because it is more than 20 times toxic as compared to the mercury.
it's not 20, it's a 30,000 times more toxic.
So for at laboratory scale, can we go for methyl mercury trial rather than the mercury? I'm just,
Yeah, we use the methyl mercury in Arabidopsis with the MERB gene. We did that, but I'm saying is methyl mercury, handling methyl mercury is extremely dangerous, extremely toxic, 30,000 times more toxic than the this metallic mercury.
Yeah.
And methyl mercury biomagnified in the food chain.
Yeah.
Even if you have the gloves, 2 or 3 layers of gloves, if a tiny drop of methyl mercury or phenyl methyl mercury goes on the skin, it absorbs so quickly and attack the nervous system. There was a case in Dartmouth College, a technician was working and had the gloves, but somehow the mercury permeate, and she was crippled and she died within a month. So it's handling methyl mercury in the lab is really, and we did, but my lab, we are not dealing with that because it's extremely toxic. And if a student, postdocs or researchers has little carelessness, it could be a disaster.
Yes, Professor. RP Singh, the last question.
Nanoparticles, we talk about the sulfur or even nitrogen nanoparticles or any nutrient nanoparticles. Are these inorganic, these molecules are toxic to animals, including human beings. Only they are not toxic to microbes and plants. So the use of these nanoparticles in agricultural system is quite hazardous to other workers who are working in agricultural system. What is your opinion on that?
Yeah, that we have the several experts on nanoparticles here, but my lab is also working on nanoparticles for last many years, and it's a double-edged sword. Basically, it goes both ways. If you are using those toxic metals as metal-based nanoparticles, it could be like there are copper kind of that. In the tomato industry, they are using the copper nanoparticles and copper sulfide and all those for controlling the wilting, tomato wilting disease, and ultimately the excessive use of that become toxic in the soil. So in this case, we selectively use non-toxic kind of nanoparticles. Sulfur is not non-toxic. Sulfur is a required nutrient for the plants. So that's where. For both. Yeah, it's for both. Sulfur is not toxic. So that's why if you have the metal base, that could be lead to the excessive use of, could lead to the toxicity, accumulation in the crops, and then also excessive accumulation in the food chain could be toxic to humans. But we are trying to use them. Like in my lab, we have used Jason and me and Bausen are using that nanosulfur and in the different forms for different metals, and it has been showing tremendous impact as a nano fertilizer, as a nano pesticide, and for food safety for reducing the toxic metals. But your point is valid. I think when you use the nanoparticles, try to make those green kind of those nanoparticles that are not toxic to the environment. That depends on when workers are doing that. If they are not using the PPE, protective equipment and all those, could be, yeah. And any of those metals, when they are using those, even copper is toxic in a higher level, it's required in small amount. People should be using precautions. Yeah. Okay, thank you very much.
We give a big hand to Professor Dhankar for a very wonderful presentation, and it shows a real phytotechnology application. Thank you. So, may I invite Upendra Kumar, Mahatma Jyoti Phule, Royal Khan University, Bareilly, for his talk on the CRISPR-Cas9 editing of OS-LSI1 and OS-LSI2 transporters for reducing silica and arsenic accumulation in rice. Dr. Upendra Kumar.
A very good afternoon to all of you. And first of all, I would like to thanks to the organizer, especially Dr. Om Prakash Dhankad and Dr. Jose Puthur, who invite me and give the chance for my talk. And this project was designed by in collaboration of Dr. Om Prakash Dhankad and funded by the Spark scheme, Ministry of Education, Government of India. And CRISPR-Cas9 editing of LSI1 and LSI2 transporter for reducing silica as well as arsenic accumulation in rice. Basically, arsenic contamination in rice agrosystem is considered as a biggest threat, especially in northern India also. Arsenic is a metalloid of group first, it is carcinogenic, and two inorganic form, that is arsenate and arsenite, and arsenite is more toxic. Okay? And many health risks of arsenic toxicity like respiratory and digestive system disorder, nephrological disorder, neurological disorder, etc. Rice can accumulate silicon to the level of up to 10 to 12% of soot dry weight, and silica is essential for high and sustainable production of rice, and it's provide the disease resistant to the plant also. And nowadays rice straw burning is a big problem in India, or basically nearby the capital region, and you see here the impact of rice burning nearby Delhi. This is the picture of India Gate, and this is in a clean days, and this is a smoke activity. Basically, for arsenic and silicon uptake, the transporters are same, that is LSI1 and LSI2. Basically, LSI1 is responsible for uptake from the soil through root hair and transfer to the endodermis, and LSI2 basically transfer endodermis to the xylem, and one more gene, that is LSI6 is responsible for xylem to xylem parenchyma, that is stored in stem. And basically our approach was to slow down both these two transporters, that is LSI1 and LSI2, not fully knocked down, and we select the target site selection in target gene using computational approach, and design the guide RNA for these two genes, and cloning into the PRGE31 vector, and finally transfer into the seed of the rice through in-planter transformation. That is our patented protocol, which is already published, not granted the patent. And then given antibiotic selection and PCR genotyping, sequencing, and finally check the expression level of that genes through RT-PCR. And we designed two guide RNA of each transporter gene, LSI1 promoter region as well as from the exon region of the both the LSI1 and LSI2. And this is the PCR genotyping of T0 plants. LSI1P, that is guide RNA from the promoter region, E from the exon region, and that is we have molecular characterize the transform lines through PCR, and sequencing results shows the insertion and deletion also in lines, and we find heterozygote as well as homozygote lines, and the transformation and mutation rate in T0 plant is around 5% to around 20%, and edited plants is in 1 to 6 plant of the lines. And this is the results of screening of Cas9 free plant, that is in T1 mutant plants, and we found some lines that is Cas9 free, and these lines having the insertion of one nucleotide, you can say here in first homozygote line in comparison to wild type, and some here is the 2 base in substitution also there, and in third line there is a deletion of 1 base and insertion of 1 base in the last lines, and deduce the LSI1 and LSI2 amino acid alignment sequence in homozygote line, and we find that the number of tDNA free plants number is very less due to the less viability of the plants in T1 generation, and we did the QRT PCR analysis of that gene. We checked the after the edited line having the lower expression of that genes or not, and we found that is around half expression in the edited lines. This is the morphological analysis of edited line. Mutation results is reduce the growth of edited lines as compared to wild type, and you can see here in plant number 4 that is almost yellowish. The chlorophyll content is very low, and grain yield is also reduced, but in one line, that is exon 2 24, plant number 24, having the yield is at par. And we did the physicochemical analysis of the edited line, and the mutated plant has faced stress leading to significant increase in antioxidant activity, and chlorophyll content was also found reduced, and these might be due to the reduction of the silica content is known as the aid for various biotic and abiotic stress due to low silica content. And we check the silicon and arsenic content in edited line. We grow the plants in hydroponically, and the effect of silicon, that is 5 millimole, and arsenic is 100 millimole. The silicon and arsenic in control and edited line, the value basically is in silicon treated lines, the silica content is lower around 33 to 70% in each line, every lines, and arsenic content is around 62 to 85% lower as compared to the wildlife plants. And the conclusion is mutated line or low silica lines can be subjected to microbial consortia for their fact decomposition of rice straw management, as they have lesser content of silica in their straw or leaves. Basically, due to high silica content, rice is taking more time for the natural degradation, and our approach was, if we reduce the silica content around 30%, we can reduce the time of natural degradation of the rice straw also, and we can solve the problem of straw burning. Second, the mutant line can be used for low arsenic lines in breeding program, and mutant line 2E34 can be used in breeding program to generate new genotype with low silicon content and high yield basically. And these are the contributor of this work, Dr. Om Prakash Dhankar and Dr. Yogita. She was my PhD student and currently is working as postdoc with Dr. Om Prakash Dhankar. And one guy that is missing here, that is Dr. Sudhir Sharma. Starting was done by Dr. Sudhir. He designed the guide RNA for all the four guide RNA from promoter as well as exon, region and clone into the vector. Thank you.
So, thank you, Dr. Upendra Kamal, for very nice presentation. You can have a couple of questions. Please.
Thank you for this lecture. If you decrease silicon in your rice line, you may change also the suberization of the Gaspari pen, and this can affect also the loading of cadmium into the xylem. So did you check about, or are you planning to check the effect on those elements and the genome of the plant in your line?
Yeah, we check the effect because if we fully knock down the gene, the plant can be converted into the climber and disease susceptibility will be there. In today's presentation, the disease susceptibility data is missing, but there is a disease susceptibility with low silicon content in our lines. Note, we are not checking the cadmium. Okay.
I can foresee some other issue like the rigidity and the rigidity of the plant if you have too much wind and when you harvest, because you have less silicon, maybe the rigidity of the plant could be affected.
Yeah. You see here. Yeah.
So, if there are no questions, we thank Professor Upendra for the wonderful presentation.
Thank you very much.
And now I leave Dr. Kundan Kumar to chair the session.
Thank you, Dr. R.D. Tripathi. So now we invite Dr. R.D. Tripathi to speak about the work, novel strategies for low arsenic rice cultivation for sustainable agriculture and environment. As we know that R.D. Tripathi has done explanatory work on arsenic, and he has command over this particular subject. He's now working as a emeritus scientist in CSIR NBRI. Over to you, sir.
So I thank Professor Dhankar and Professor Pathur for inviting me for showing some of my work which I did at NBRI, and we had a collaboration with Professor Dhankar, Professor R.P. Singh and many. So, arsenic is a problem, and it is, I think many previous speakers have already given so much background, which makes myself to cut some time. But I'd like to say that arsenic is a problem through various aspects. It's in many countries, it is a problem because anthropogenic. Well, in India and Bangladesh and many other countries, it is a problem of the groundwater, and we have we are exposed to arsenic through the groundwater, and then we. And through that rice is a crop which is irrigated by the groundwater, and it requires much more wetland condition in order to have higher productivity. And therefore, and because of the highly expressed pathway as compared to wheat and maize, rice has one order of magnitude more expressed the silicon pathway. And it is because of this over-expressed pathway, silica pathway, that arsenic gets into the, because of the growing conditions, into the rice, and it goes into the grain. And rice causes arsenic accumulation causes different kinds of diseases. Many are carcinogenic, and just to tell you that one of the compounds, arsenic trioxide, is a chemotherapy drug for one of the leukemia. So through this presentation, you will find that the arsenic, and sometime I will be talking about the selenium also, which is a antagonist of arsenic, and we both they cause cancer, and they cure the cancer also. So that is important. So I think we have already listened about these things, so I will skip. And we can, because of this, there are, because of the repeated loading of arsenic through groundwater irrigation, and different countries have the range of arsenic contamination in the soils. In India, 3.5 to 460 ppm of arsenic. Argentina, 0.8 to 33 ppm. In China, 0.01 to 26 kV. In India, we have in some states like Hyderabad and Telangana, we have anthropogenic arsenic because of it is coming through pesticides and herbicides besides the groundwater. So in Pakistan, we have 8.6 to 266 ppm. In USA, 2.6 to 417 ppm. So in US also, I think it is the arsenic herbicide and pesticides, they are used as the defoliant for cotton, and they contaminate the soil also. In Bangladesh, the hottest spot in the South Asia. So there is a lot of problem, and the problem we have about 4 to 83 ppm of arsenic in the soil. Nepal about 6.1 to 16.7, Vietnam 6 to 35 ppm. Iran, we have very high problem of arsenic in the soil, and the South Africa it is up to 75.3 ppm. So these are the different states which are contaminated with arsenic, and we can find that this in the Chandigarh, and in the Bihar, in the Uttar Pradesh, West Bengal is highly contaminated with arsenic. And so I think this kind of picture has been shown by the earlier presenters also. You can see different kind of. The one thing important that already it's an old site, and you can find that the chicken meat which is used in the McDonald's, they were also containing the arsenic levels, and FDA had admitted it, and that was the news. So in India, we have surveyed different blocks of arsenic contaminated blocks, and you can find that the red region is showing this the groundwater contamination more than 50 microgram per liter, while the limit for the WHO limit for the drinking water is 10 microgram per liter, and so EPL limits are even lesser. So you have different shades of dark green to yellowish green, then green, and then so arsenic contamination more than 50 microgram per liter, arsenic 10 to 50, then arsenic 3 to 50, and in the green is almost very safe. And all the red blocks in the they have found that they have arsenic contamination more than 50 microgram per liter, and it is because of these. For example, in the Bangladesh and in West Bengal, we have all the three crops of rice: the summer rice, the winter rice, and in between they have an house rice also. So they are highly exposed, and most of the productive varieties of rice, they are grown in the summer season. when they require more irrigation water and therefore more loading of arsenic. So this is the very, I think we talked earlier, just talked about this. We are the phosphate transporter arsenic enters, arsenic enters to the aquaporine transporters, and we were discussing about this ACCR. This is absent in the flowering plants, and we were discussing about that. So it is present in the fern family, and have been found in the Pteris Tata, and it has capacity to directly, since it is a plant. Pteris Tata is a plant which can accumulate lot of arsenic, and then it can look very healthy. So it doesn't have a phytokeratin type detoxifying system. Therefore, it can directly it has this transporter ACR3, which is an antipoter. So that ABCC is a transporter which transport the phytokeratin complexes in the plants. The phytokeratins are have different these are the metal binding peptides. They are the polymers of glutathione, and they can be in the plants. They can be repeated n times the value of n, maybe 2 to 11. So you have a polymer of glutathione, and because of the high cysteine residues, they can bind to the arsenic, or they can bind to the mercury also, and they can bind to the cadmium also. So that is the most prevalent detoxifying system in the plant. So in fact, in my presentation, I will be telling you about some of the varieties which have been identified by the so we have speak about more than 300 varieties of rice, and some of the varieties they were in the remote areas. Some were from the International Rice Research Institute, Philippines, and so from the group we have cultivating different arsenic contaminated regions of West Bengal, so Gayagata, Chinsura, Durgapur, Purvastali, Beldanga, Birnagar. Birnagar is very close to Bangladesh border, and I can tell you that we have traveled to those places, and people don't marry their daughters, which are the villages are nearby Bangladesh because of reason, but there are arsenic contaminated signs, and they think that their daughter ultimately she can lead a very bad life there, and she can die of the arsenic contamination. So then they don't marry in those bordering villages. So according to WHO, 2 micrograms per kg body weight, like medicines we take per kg body weight. So and a average body weight person, say 60 kg body weight person, and which consumes 420 gram of rice as a subsistence diet. In Bangladesh and Bengal, they have subsistence diet, lunch rice, dinner rice. So in that case, all these varieties which have if you calculate from this data, you find that the varieties we have 286 micrograms per kg of rice is safe for eating, even as a subsistence diet. So all these varieties have been identified. They are popular varieties, productive varieties, and this variety CL179.2, that's CSCR MBRI, that in fact it was developed as a new new variety, low grain arsenic variety, and this variety was approved by State Variety Release Committee and then the Central ICR Committee in 2019. And I'm very happy to tell you that these more than 150 farmers were growing these varieties in their fields, and this has been recommended by the government of West Bengal. This CL17942 test. So this is a popular name. Mukta Sri is given by the Chief Minister, Mrs. Mamata Banerjee. So Mukta Sri, it is free from arsenic, but that is not the truth. The truth is it contains the safe level of arsenic. So these are the Mukta Sri variety, and this looks like how it looks. It's a basmati type variety, and this is a selection from a traditional basmati type variety, Karnal local. So we could do this work in collaboration with Rice Research Institute, West Bengal, and at MBRI. So this variety is very safe and traditional basmati type variety. And as we are discussing that arsenic accumulation leads to decline in some of the trace elements which are required by the plant and the human being both. And these are the arsenic level, the low arsenic variety which have been identified in the West Bengal. You can see this part number second is the Gupta sequence. And you see, look at the yellow bars. This is the marker variety. This is called the BRG12. So it accumulates a lot of arsenic. You cannot eat that variety. But in such programs, we need to pick up a marker variety. So you can see that marker variety is the BGR12. It's accumulating a lot of arsenic. But you can see the Mukta C at different places is the lowest quantity of arsenic. And we can see that when the variety which accumulates a lot of arsenic, you have a decline in the trace elements which are required for the human body. For example, you can see that this iron level is very much low. So here in the Gupta, you can see the iron level is very high. So the variety which accumulates low arsenic accumulates higher level of arsenic. So this is the summer data, and this is the second one. This is the green data, so wetland rice. So you can see that in both the conditions, when this variety has a low level of arsenic, at the same time, this is how it grows in the fields, and it accumulates a high amount of iron. So because in those areas, in fact, the children and the pregnant women have the deficiency of iron in those. So this variety is very useful. This is a full view of some of the varieties that are growing, including the Muktasri. So we have another approach also. We have a transgenic approach, and in which we have used two genes. One is phytochlorine synthase, and the other is arsenite methyl transferase from a fungus which was growing in the arsenic contaminated soils of West Bengal. So I will tell you that there is a plant Ceratophyllum demersum, which is a plant which accumulates a lot of arsenic. I don't know, some of you must have seen this. Is a rootless plant. It's just like hydrilla. It's thinner than a hydrilla plant, and it's a rootless plant. So one of my students, Dr. Siva Misa, worked on this plant, and she's an associate professor in the chemistry department in the Lucknow University. And we made the transgenic lines, and we made this. You can find that the plant was very useful. The transgenic plants gave a very useful high amount of arsenic. This is the sequence, there is 503 amino acids, and we had made the transgenics, and these are the rice transgenic plant, particularly this is plant, and you can see that in the grain it has about 60% decline in the arsenic content, and we used a CPME promoter, and you can see that this low arsenic grain arsenic is because of the high sequestration in the roots. At the same time, you can see that this variety is having the highest tiller number, highest grain yield. So just I tell you this Vesta Dicala abratica is a scomatis seed fungus, and they isolated from the soils, and it has a small gene, arsenite methyl transferase, and when we made the transient lines, so you can see these are the control. This gene is microbial origin, so there is no expression in the wild type, but you can see that more than 10-fold increase in the utilization of arsenic. So arsenic is, arsenite is the most toxic form of arsenic, and all the three hydroxyl groups which are present, when they are methylated, they form a trimethyl arsine, which is volatile. So it escapes, and it has no toxicity. So I think this gene is very, very good from the phytoremediation point, and from the decreasing the load of arsenic in rice. You can see that in the root fruits and in the pollen grains also there is more than 50% variation in the when we make transgenic transcript. Western Dicala, Abratiata, arsenic methyltransferase. So just I'm going to finish. So because selenium is used as an antidote of arsenic, and they use the selenium because they share the same pathway. Because see, this is the red line is arsenic, and there is a big decline in the selenium. So when selenium is less, arsenic is more. So that's the antagonistic nature of the. So just experimentally proved that when you use selenium in the fruit, there is a 50% reduction in the sugar, 54% reduction in the grain, 40% reduction. So we conclude that screening and selection of multi-location field trials is low-grain arsenic cultivars resulting in the development of a new rice variety, which is known as the Muktasi, which is recommended for arsenic contaminated regions in West Bengal and many other states in India. And aquatic plant was a good source of phytotechnology in theazine, and heterologous expression this is methane in rice leads to lower arsenic accumulation in the grain, to high arsenic concentration in the root. Transgenic rice plant expressing Western Dicala and Ratka arsenic methyltransferase with a high tolerance, high arsenic concentration, and low accumulation rice seed, safer for human consumption. Selenium supplementation has been found to minimize arsenic accumulation, toxicity, and improve the nutritional quality of rice. So, if you increase the selenium content, selenium is a trace element. We all require 90 microgram of selenium every day. At the same time, it can prohibit the arsenic accumulation in the grain. So, it was not possible to carry out this research to the economic side, Dr. Devasya Chakravarti, Prabhu Trivedi, Dr. Linda Singh, Dr. Vivek Pandey, Sanjay Divri, Shekhar Malik, Siva Mishra, Daniel Shukla, Mudhu Shukla, Priti Tripathi, Madhu Sri, and you can say, we collaborated and we had a large, good number of publications. So it is the development of a variety which was a selection variety because of the production right research mission in Silsila, the West Bengal, division Dikari and Mazar. So that's all. Thank you very much.
Thank you, Dr. Tripathi. It was very elaborated presentation on arsenic contamination in different parts of India as well as remediation purposes. So few questions, maybe one or two questions we can take because we already exceeded some time. One or two questions. Yeah.
Very nice talk, Dr. Tripathi. And I want to just ask you one thing. With this VARS and methyltransferase, so when you said it's making a trimethyl arsine, so what happened to that trimethyl arsine?
Trimethyl arsine is I think many thousand times less toxic than arsenic.
Yeah, but what happened when
in the atmosphere? Yeah, it goes into the air.
But it come back though.
After all, there is a biogeochemical cycling of the arsenic, and you have worked in your postdoc, a lot of work in the biogeochemical cycling of mercury. So you cannot prohibit the cycle, it goes on.
Because metal is metal, it will be steady, it will be coming down.
Yeah. That is the issue, but in that way you can say that people say that the food plants cannot be used as a phytoremediator plants. But in this particular case, when you can, even rice plant you can use as a phytoremediator plant because it volatilizes. But the gene is in the this pathway, it is called a challenger pathway, it is operational in the microorganisms only. So you can find this arsenic methyltransferase only in the microbes. And this gene is absent in the flowering plants.
Thank you.
So thank you, Dr. Tripathi. Thank you. Now I would like to invite Jyoti Mathur, Dr. Jyoti Mathur. She is from Banasthali University, Rajasthan. Her topic of presentation is phytoremediation efficiency of hyper-accumulating plant species, Helianthus anus and Tagetes erecta for mitigation of heavy metals from industrially contaminated soil. Over to you, Dr. Mathur.
Yes, ma'am.
Okay. Okay, so good afternoon, everybody. And I would like to thank Professor Dhankar sir and also Professor Jos Puthu sir, organizing secretary of this conference, for providing me the opportunity to speak over here. Since morning, we are actually talking about phytoremediation, listening so many plants which has been actually worked for the phytoremediation, in fact. And we know up to now, I think every audience, including the students, I would specifically emphasize to the students that they should focus on, they should be initially capable of selecting the problems on which they should work. And researchers are in fact doing their research, but the students, the audience who are working present over here, I could see since morning they are not responding. So that's why I am specifically emphasizing this point. So continuing to my research topic, which is phytoremediation of arsenic hyperaccumulating plant species, which I used Helianthus annuus and Tagetes erecta. Research problem, basically origin of the research problem is more important. We already know that there are so many sources of heavy metals. We already discussed so many sources, like so many industries, mining in, area, another area. And in fact, in the morning, Dr. Lees, she has also emphasized on deterioration and accumulation of TNT from the site. So I also initially worked on what are the plants which can be explored for phytoremediation. But I just switched to from the collection site, from the industrially contaminated site. So laboratory work, it was the preliminary work to optimize all the conditions for phytoremediation. But I started then, here only I am just presenting that how the soil first is analyzed from the contaminated area. We actually collected the soil sample from various contaminated site and then proceeded for phytoremediation. This is the only difference from the previous talk to mine. So these are the sources of heavy metals what is mentioned over here. So I am actually focusing on various industries like paper industry, plastic industries, battery, effluents, textile, paint and pigment industries. They are actually having so much of effluents which is coming out of the industry, and in a way they are contaminating our environment. Those industries, even if the Central Pollution Control Board has given some permissible limit that they can have this much only, but the reality is nearby industrial region, if you go to those sites where dye industry is there are so many manufacturing industries, pulp and paper, you can find lots of amount of heavy metals in the soil as well as in the water to the nearby area. I mean, even it's pond. And the most hazardous part is the farmers which are not aware that how heavy metal is creating toxicity to the human beings, how these are actually being entering to the food chain. They are unknowingly using those water only which is coming out of the industry, and somehow it is being accumulated in the pond nearby area. So they are using as a part of irrigation. We personally visited to the site and seen that and taken the samples from there and talked to the farmers. They are using those water to irrigate the vegetables, to grow the plants, and in fact it is a part of their occupation. That is the main source of income for them, so they don't worry about that. So this is the major problem. We as a science person are very much aware, but how much those persons which are actually working on that land, they should be aware of this particular problem. So these heavy metals, here what I worked on, these are the experimental plants which I worked on. One is Helianthus annuus, sunflower, Tagetes erecta, Medicago sativa, and Canna indica. Just recently I have taken to remediate many of the metallic species. So mainly these plants, soil sampling from various industrial sites, that was the first and foremost important things, and then soil sample analysis how much amount of the heavy metals these soil samples are actually containing. And in the pilot experiment, initially we have already tested that, as my student Sanika, she has also presented her work, and other students, they worked already upon their highly hyperaccumulating plantar species. We have many published paper that shows that more than 700 ppm of the various zinc, cadmium, arsenic, mercury, they are actually being accumulated by these plant species. So after sampling, you can see the sampling site. Definitely for any experiment, we have to take some control soil sample. So from Krishi Vigyan Kendra, which is inside the campus of the university, we have taken the control soil sample. Along with that, several factory visited some Uttarakhand district, some UP, Central India, UP, and even Rajasthan. Several industrial sites, you can see dye factory, textile industries, plastic, even oil mills, and paper industries, iron factory, leather industries. We have visited the nearby regions and collected the soil sample, and after that analyzing. So these are the sample side, this kind of effluents that is actually coming out from the industries. So after soil sample collections from the various industries, we just planted our phytoremediator plant, which has been selected onto this soil sample only collected from industrial regions. After that, we analyzed that how much of heavy metal can be accumulated. In fact, what is the realistic thing that after contamination, how much amount of the heavy metal that can be removed from these plant species. So there are so much of amount of heavy metal which has been reported in like lead, more than 104 ppm, 185 ppm, even arsenic, even cadmium, zinc, mercury, so much of amount. This table shows the characterization of soil samples from various industries. So this is the outline of the whole lot of experiment we carried out after planting the this hyperaccumulating species, Helianthus as well as Tagetes, inside the industrially contaminated soil. We analyze that whether they are negative effect onto the plant when we are actually using. See, when you're using an experimental level, then 1 or 2 metals we are artificially incorporating. But when we are collecting the soil sample from original industrial zone, then it will be multi-contaminated soil. So then your selected plant, whether it is surviving over there or not, we have to definitely check it out. So after this, what we did, we grown these plant species inside the soil. It is the soil from various industrial region labeled as S1, S2, S3 up to different. So first one, Helianthus, very well surviving, and Tagetes also very well surviving in maximum of the contaminated soil. Okay, so uptake efficiency of heavy metals that has been shown in this particular graph, that it is very highly accumulating species, Helianthus as well as Tagetes. So the graph thus goes up and down, that shows that depending upon the metal contaminants in the soil, the accumulation by the Tagetes as well as Helianthus that has been taken up for the lead, cadmium, copper, zinc, and other metallic species. So this graph again showing the accumulation of cadmium and arsenic by Helianthus plant species. After planting, not only morphological, we have to go for biochemical and other responsive factors. Antioxidant enzymes, they are definitely a part of defense mechanism in the plant. So proline and polyphenol content analysis that shows that how the plant is actually surviving, defending against metallic stress. Since we are growing the plants inside heavy metal contamination, so we have to check that what are the genes, what are the factors which are responsive for against the defense system. So proline playing very important role in the defense system, polyphenol again. For glutathione reductase, GR, GSH and PCS, Many of the researchers, Dharkan sir, even Tripathi sir, just recently talked about DSH and GR activity. So definitely these enzymes need to be quantified. What is the level of expression? What are whether these enzymes are expressing at a higher level under the stress condition? So yes, we found that these enzymes are expressing. The graph goes up when they are under the metallic stress. Another antioxidant that is the similar one, we have also gone for the SDS page, where we are going for metallothionein genes proteins expression. So protein level metallothionein specific proteins that has been isolated, and that through SDS page that has been quantified. The level of metallothionein protein also, which is going up in these specific plants which are tolerant to heavy metal. Now, I think you all are aware that one of the plant which I have taken for the phytoremediation, and many of the papers, including ours, that reported the helianthus as one of the highly accumulating plant species, but still you might have heard that sunflower is actually yielding sunflower oil too. And it is one of the obvious questions that we should ask, whether the oil goes to this, whether oil contains heavy metals or not. So here in this formula, we have already analyzed the seeds of the helianthus growing in the contaminated area. So the seeds does not contain any amount of heavy metal. That shows the very, very less translocation factor from root to shoot, and then shoot to flower, less translocation factor. So it is a kind of rhizofiltration, which is one of the method for a phytoremediation process. So it means the translocation from root to up to the flower, and in fact to the seed, is very, very less. That can be used as a phytoremediated species. So again, the work is being authenticated by microscopic analysis, light microscope followed by fluorescence microscope and a scanning electron microscope, along with transmission electron microscope, to localize where the heavy metals are actually going inside the anatomical or ultrastructural part of the plant. So we could localize these heavy metals inside the root stem if we go to the ultrastructural view. So through light microscope and through fluorescence microscope, we have some specific fluorescence dye which binds to the heavy metals. And then even through the FESEM EDX profile, which shows that it contains the heavy metal in a scanning electron microscope. We could see through transmission electron microscope, through ultra thin sections, TEM, that shows also that there are less anatomical changes, microscopic changes, and localization of these heavy metals inside the plant parts. So other than that, molecular expressions, gene expression, so through RNA isolation and qRT-PCR, we proceeded with this qRT-PCR and just checked this expression of GSH, and again along with the relative expression, we got the higher fold changes in the gene expressions when the plants are actually growing under the stress condition as compared to those which are not under the stress conditions. So these are the approach which actually authenticated, and this is well proved that these plants can be used as a phytoremediated species. Along with that, we found some of the hydrocarbons also, since oil factories are there, some petroleum factories are also there near Jaipur. So we collected some soil sample and found some hydrocarbons associated with that. And just one or two slide I would like to show that these plants are even capable of accumulating pyrene, anthracene, and some of the PAH, polyaromatic hydrocarbons from the soil. Okay, so although up to a certain limit, not the higher one, since hydrocarbons are really toxic even for the plant species, so up to a certain limit they are capable of accumulating and phytoremediating these species. So there are so many things to discuss, and since the time is actually the warning bell is for me, this slide again shows that how the hydrocarbons PAH, and these are actually being localized inside the plant tissues. In conclusion, I would like to say that these selected plant species, Helianthus, Anoas, Tagetes erecta, even some of the Yorcana indica, these plant species are capable of accumulating. The main thing is implementation onto the site of contamination. there should be a lab to land technology to transfer this, whatever we are working inside the laboratory, that has to be transferred to the original site. Then only our research is successful. So we tried this approach. Initially we have done so many experiments in authenticating them, and then now we have already taken a site to the contaminated land and trying to work like she has worked on decontamination of TNT. In the morning she just explained about that. So we have already taken some site, talk to the farmers, talk to the peoples who are residing nearby the regions, and taken the slide to plant these trees for implementation of phytoremediation. These contaminants, in fact heavy metals like that. Thank you so much. These are my publications. We have published these works in the manuscripts, environmental science and pollution research and others. And this is my research team. Without my research team, I have completed more than 9 and 10 students as PhD students. They have been awarded, and right now they are all working with me. Without their efforts, it's not possible for me to present my work over there. Here. Thank you so much. Thank you so much for your patience.
Thank you, Dr. Mathur. It was a nice presentation. So very few questions we can take, 1 or 2 questions maximum.
So here is a question from me, couple of questions. Well, it is interesting to know that there is rhizospheric exclusion that is happening with respect to these plants that you have analyzed. Out of curiosity, did you analyze the flowers, whether the flowers were able to uptake the heavy metals?
That was the only part. Thank you. I'm very happy that student, you're a student.
I'm a faculty.
Okay, oh, sorry. So yes, we analyzed, and in fact it is a part of analysis. Root, stem, leaves and flower. Everything has to be analyzed. Then only you can come to the conclusion that it is a phytoregulator.
Okay. Another question that adds to it, with respect to the light microscope images that you have shown, the anatomical sections, you have pinpointedly said that these are the heavy metals. And then you said that under the fluorescence microscope, you have used very specific dyes. Could you please specify the dyes you have mentioned?
Yeah, it's a kind of zirconal and acridine dye. There are so many fluorescence dye that can be used for
but
that.
Do you really think acridine orange would be able to localize the heavy metals?
For light microscopy, we do not require. We can compare with the control one and the treated one.
So
right?
you mean
we
to
always
say,
have a we always used to have a control with us, and through control we can compare what are the anatomical changes. Point number 1. And for fluorescence, we have so many other dyes. For lead and cadmium, some specific dye is there. For arsenic, there is blackening, which is compared with the control one, and again it is validated by SAM, where SAM EDX that shows the arsenic inside.
So we are running out of time, so maybe you can discuss the further things on in the tea break or so.
Okay.
Yeah, thank you, Dr. Mathur. So now I think it's an online presentation by Khalid Stani Kazal. The presentation is on phytoremediation of zinc and lead from polluted soils using sunflower. Yeah, Dr. Khalid, can you?
You may on your video, sir.
Yeah, we can.
Hello, good evening.
Yeah, you go ahead. Yeah.
You are audible. You are audible. Hello. And you are good to go. Yeah. Am I clear?
Yes, hello? You are clear. You can go ahead. You can continue with your presentation.
Okay, thank you so much. This is Khalid speaking from Kabul, Afghanistan, from Kabul University. Actually, I have done my MSc in India from 2017 to 2019 through ICAR scholarship. Currently, I'm a lecturer in Kabul University in Department of Forestry and Natural Resource Management. My research was on excuse me, sir, something happening. On phytoremediation of zinc and lead from polluted soils. This is my table of contents: introduction, objectives, methodology, result in discussion and references, because I made my presentation not complicated, easy. All respected professors, assistant professors are discussed the issue. Just as we know, phytoremediation is one of the cost-effective and easy methods for removing of heavy metals. I want to directly jump to my objectives. Sunflower is a plant grown in late and zinc-contaminated soils, underwent pot trials to examine its biomass and to undermine how well may remove the metals. And first, my methodology was soil preparation and pot experiment. In figure 1, pots cleaning its preparation for starting the experiments, and then in Pantnagar University, sieved soils ready for conducting pots experiment in glass house and CBSH College. And the soil was collected from the Crop Research Center, GB Pant, the University of Agriculture and Technology. For pot experiment, collected soil were air dried in the shade of the field, and stones, plant debris and pebbles were removed by hand, sieving of soil and mixing thoroughly to make a soil homogeneous for the experiment. Before starting my experiment, I did the tests of physicochemical characteristics of the studied soil, such as pH, electrical conductivity, organic carbon, total lead, total zinc, and particle size distribution and texture classification. I did it in College of Agriculture in our department as well. By this two heavy metals, I conduct the experiment by AAS, atomic absorption spectrophotometer. Selection of a plant. The phytoremediation experiment was conducted on attractive and quite economic plant, Helenthus anus, in pots. Sunflower, as we know, the sunflower is a widespread plant species which grows very easily. Sunflower is tolerant to heavy metals. The experiment design consisted of 33 pots, 2 heavy metals, and 10 into 10 treatment, and with 3 replications. The pots were placed and arranged in the glass house of the College of Basic Science and Humanities, GBPUNT. And soil analysis, the physical and chemical characteristics of the soil were initially investigated in the Department of Soil Science, College of Agriculture, India, and pH of the soil was measured by digital pH meter, carbon and soil through the weekly and black method. And after starting the planting during 9 weeks, I had recorded the data, planting sunflower seeds in controlled and treated soil in controlled greenhouse, glass house, and there was no germination was observed, and 3 seeds were planted in each pot, added the tap water, and week 2 inspecting for seed germination and checking for water, which I observed 1 or 2 seeds were germinated in each plastic pot, added again tap water. In week 3 inspections, and also week 4, I did again inspection for any changes in phenotype in its growth. Seedlings of sunflower in high lead polluted soil were shorter as compared to low and normal concentration. Again, I added the tap water 2 times a week. Week 5 inspection for any changes. 1 or 2 plants of sunflower were died in lead contaminated pots. Added tap water 3 times a week. Week 6 inspection for any changes in the phenotype of sunflower plants. Again, added the tap water 3 times a week, having a good and healthy growth in low concentration as compared to high concentration of lead in zinc contaminated plants. They were healthier as compared to lead. Week 7, checking and monitoring of experimental pots in the glass house, added tap water 3 times a week, good and healthy growth of sunflower plants in zinc treated and untreated soil as compared to high treated with lead. In week 8, checking and monitoring sunflowers plant and healthy growth of sunflower plant in normal and treated with zinc as compared to lead polluted soil. And week 9, so this is my experiment with figure 3, 4 weeks experimental pots showing growth of sunflower plant with different concentration of lead. in glass house, 8 weeks. This is 8 weeks experimental pots showing growth of sunflower plant with different concentration of zinc and lead inside the glass house. This is 9 weeks showing growth of sunflower plants with different concentration of zinc and lead inside glass house. So plant harvesting analysis, after 9 weeks of growth, sunflower plants were carefully removed from their pots. After dividing the shoots in rows, their width and length of each were recorded to get rid of dirt and other particles. All the plants were pots were thoroughly cleaned with tap water. After that, it was dried at 70 centigrade after being cleansed twice with double distilled water. And at the end, atomic absorption spectrophotometer was used to measure the dry matter of these sunflower biomass. So this is digested sunflower biomass filtration. It's preparation for AA analysis, and digested and filtered sunflower root ready for AA. This is at College of Department of Environmental Science at Pantnagar University. The data which I received was with increase of lead, the average shoot was decreased, and again average shoot dry weight with increase of lead it decreased, as well as zinc and table 4 fresh and dry weight of sunflower root cultivated in treated soil with different concentration of, again average root fresh weight it's also increased. So let me show it in a graphical representation. This is my whole figures shoot fresh weight of sunflower cultivated in treated soil with different concentration of lead and zinc. In figure 2, shoot dry weight of sunflower cultivated in treated soil with different concentration of lead and zinc. Again, figure 3, fresh weight of sunflower root cultivated in treated soil with different concentration of lead and zinc. In figure 6, root length of sunflower cultivated in treated soil with different concentration of lead and zinc. Again, figure 7, different concentration of lead and zinc in sunflower, which has the impact. So, according to the findings, the fresh and dry weights of the developing plants gradually reduced as the amount of heavy metals in the soil increased. The fresh weight of the shoot and root were decreased by the application of 835 milligram per kg soil of lead and 598 milligram per kg zinc. The maximum concentration of lead and zinc in the shoot 146.27 and 277.83 milligram per kg, and rods were 589.77 and 287.97 milligram per kg respectively was recorded in the plant cultivated and contaminated soil. So the root growing in soil treated with 835 milligram per kg had the maximum bioaccumulation of lead. The control soil had the longest plant shoot and root lengths, averaging 75.6 centimeter and 9.5 centimeter respectively. Under the soil of lead concentration of 835 milligram per kg soil, the shortest plants had shoots that measured 58.5 centimeter and roots that measured 6 centimeter. Similarly, when the soil was treated with 598 milligram per kg soil zinc, the plant shoot and root decreased dramatically by 55.67 centimeter and 7 centimeter respectively. As a result, the study shows that sunflower plants were better at absorbing lead and zinc, and we recommended using them to clean up soil that has been contaminated with these metals. So that's why I made my presentation brief and
You, Dr. Khalid, for a very nice presentation, but because of the time limitation, I think we cannot take any further discussions. So there were 5 presentations.
That's why I made my presentation so brief.
Thank you so much. So there are 5 presentations, and they use the different kind of plants, energy plants to crop plants to ornamental plants, telling different strategies of phytotechnologies for food safety and phytoremediation. So we thank all the speakers, and I'm informed by Professor Dhankar. He has to make some announcements.
Yes, thank you to all the presenters, but the other group has already finished. For half an hour, they've been waiting for the photographs for us. So right away, we will be rushing to the side building on the balcony for the photographs, please.
Sure.
Thank you. And then there will be buses to go to the botanical garden right away. Thank you. You can have a bio breaks if you want a little bit.
Nice job. Nice job. Thank you. Thank you so much.